Good evening, good evening, good evening, good evening, good evening, NEET warriors. I hope now I am clearly visible and audible. Everyone, I hope now I am clearly visible and audible, everybody. So, how are you? How are you, Arsh? How are you, Henry? I see these names. I guess these names I used to see in my J channel. But still, have you given your NEET attempt or what, beta? So, good evening, everybody. Let me just refresh that comment screen because after that, we'll be soon starting the session for which we all have been waiting, I guess, since to you it seems ages, but no, beta. This is just a phase of life, and however it has been, it was just a phase of your life. So, future doctors, let us kill and let us explore the analysis part, that how was it, and all the future doctors, the NEET 2027 aspirants, NEET 2028 aspirants, the analysis is going to be really crucial for you because we have to judge and we have to actually anticipate what NTA is finding interest in. Now, as a whole, beta, bio was very easy. Yes, we have also now madam will be coming and giving you the complete analysis regarding bio. I guess the paper, we have just got the rough content of the paper. Let me show you. But yes, it is just getting assembled, but we wanted to meet you, to meet each and every one of you. So let us explore the questions. Now this is, I guess, biology. So let's move to our area of your NEET exam, which is definitely chemistry. So this is all your biology question. Let me directly move. Yes, biology was beta. Quite doable. If you are very much religious with your content, then it is always easy to attempt your paper. Many of them will say, "Arey, paper is difficult." If you are actually aware with your content, then what is difficult? If you do not remember any fact of NCRT which should be there in your head, then it is difficult. If you have done questions of that sort, how can you say the paper is difficult? Beta, it is all dependent upon the level of revision that you guys have. And yes, it's a very crucial phase of your life because for a JE aspirant, getting a engineering degree from a private college is okay, but getting an MBBS from a private medical college is impossible because obviously it's huge, the amount that you in fact your parents have to pay. So yes, you have to be religious with your content, and if you do so, now let us have some analysis of the chemistry because madam will be joining us soon for the part of biology. Yes, I guess these are our chemistry questions. So chemistry, I guess it is starting from this is your physics 36. Now, yes, from here we have started our chemistry. So this is where I will just we'll be switching rounds because you can obviously see, sir, I cannot attempt paper with this white because this is the actual paper of one of our student. So this is the paper from which we have started chemistry. So now, how you should have an habit of attempting your paper, beta? Just read the question. Now, sir, this is question is having option A, option B, option C, option C. You can clearly see, sir, this is isopropyl alcohol forming phenol. This is what, sir, NCRT, alcohol, isopropyl benzene forming phenol is the basic conversion of cumene into phenol. So cumene into phenol is done by what? Sir, you have to first oxidize. That means A is 2. So if A is 2, yes, this is directly a paper from a student. So you can see A is 2 is option 2 also, 3 also, 4 also. Then now go to elimination. So you should attempt these papers. Now the paper is just getting assembled properly, beta, because obviously it will be very difficult to attempt such questions. But if you try and attempt such questions properly, now if you say, sir, this is difficult, then if you have not read your NCRT, then it will be a difficult paper for you. So let me check whether the yes, so the papers are still getting assembled. By the time what we just start attempting this one, beta. Chalo, let us attempt this one. What does this question say? I have isopropyl benzene. Now let me just zoom in this for you. Now I have this isopropyl benzene. This isopropyl benzene is converting into alcohol. Sir, what happens? First oxidation take place. Then after oxidation, sir, rearrangement take place and you form phenol and corresponding acetone. So that means A is definitely 2. So A is definitely 2. That means you have eliminated one option. After that, sir, this is, I guess this is acetic acid, CH3COOH. Acetic acid is converting into ethanol. Sir, this is reduction. So choose a reducing agent. Where do you find a reducing agent? Sir, Acha, it is not evident from the options. Chalo. So by the time, okay, we'll just wait for the clear version of the paper to come to us. But yes, let us explore the chapters from which we are getting this paper. Now, beta, this is a question from, I will say alcohol, phenol, or ether, and to this question I will rather say it's a moderate question. You should not attempt such question in the first go because it is having levels. Now, the second question is from, sir, again, amines. First you are having chlorination in presence of sunlight, so it will be allylic substitution. After that I am reacting it with amine, so amine will make it amide, and that amide is undergoing hydrolysis, so acidic hydrolysis will give you the corresponding product. But yes, again to this question I'll say moderate. A question from equilibrium. In a qualitative analysis you are sir, generally ionic equilibrium question always associates some numerical in it. So you are given a question, you are given a equilibrium where K is mentioned as 4 into 10 to the power minus 2. Now the hydroxide is giving you BiO minus along with O minus and OH minus, and the K for the reaction is 4 into 10 to the power minus 12. Okay, it is telling you you have to calculate the pH when the following equilibrium exists. So the K value is given to you. So K will be concentration of products upon reactant. Sir, because it's a strong electrolyte, because it's a weak electrolyte, so you can obviously say K will be equal to C alpha square. K will be equal to C alpha square. What will be the concentration of OH minus? Sir, it will be C alpha. How can you calculate alpha? Just put the value. Alpha will be equal to under root of Ka upon C. So sir, this is clearly under root of Ka into C. Ka value is given, C value is given. You just have to put the minus of log of this will give you the value of pOH. So what will be the value of pH? Sir, 14 minus pOH will be the value of pH. Now you can obviously do the calculation, and we will do the calculation. Just we are waiting for the clearer version of the paper to be with us so that we can attempt those things in per slide each. But yes, I would rather say chemistry was also moderate. I mean, with the questions that I'm seeing right now, obviously you should also start attempting those questions. The next question is from electrochemistry, or rather I would rather say mole concept. When 1 decimeter cube, again they use this logic n number of time, decimeter cube is nothing but 1 liter. So when 1 liter of CO2 gas is passed over red hot coke, the volume of gaseous mixture after complete reaction at STP becomes 1.4 liter. So coke, as in carbon solid, you are passing it over CO2. When 1 liter of CO2 is passed over coke, so C plus CO2, C plus CO2, whatever product you are forming, after reaction it is saying it becomes 1.4 liter. That means the volume is increasing. That means that more of the gases are produced. So again, you have to know the inorganic side of the question also. Now that inorganic is mixed with physical chemistry logic. So this is what I was telling you before also, that NTA is doing this. They are mixing chapters. So technically it used to be done by good questions, that you're mixing inorganic concept with physical logics. So yes, to this question also I would rather say it is a moderate question. So for example, see how they are challenging the psychic of a student. They are putting all the questions in the initial phase so that you get stuck. So leave those questions and move to the and move to the doable ones, because obviously once we'll start having an idea of what are the questions which we can easily and clearly attempt, it will be very easy for us to do it. So okay, I'm just waiting for a clearer version of the, okay, finally we are, these questions are, I guess they are just getting typed. And okay, so your biology madam is ready, Ambika ma'am is ready for you with the content. And okay, chalo, so Ambika ma'am, over to you. I'll be coming with the slides beta for discussion of chemistry.
Guys, am I audible, visible? Am I audible, visible? Guys, am I audible, visible? Am I audible, visible? Hi everyone. Hi everyone. Am I audible and visible? Yes, everybody. Yes, everybody. I think I'm perfectly, yes, I'm audible and visible. Guys, the paper is very easy, right? When it comes to the biology part, it is very easy. First of all, tell me, how are you? Done with your paper? Have something done with your paper? How are you all? Feeling good? Feeling positive? Guys, paper is very easy. As per biology, I can tell you, I was just analyzing the paper, right? And you all have interacted with the Gaurav sir. I was analyzing the paper. Paper is very, very easy. If you guys have attended our last 1 week, last 1 week, I'm saying, right? I am pretty sure you were able to solve it, because paper is direct, right? I still know from my predictor session, beta, many questions that this type of question will come, that type of question will come, that type of questions were also there in the paper, right? So I hope you guys have done well, right? You are going to get the detailed solution plus accurate solution here on PW NEET English. So let's see, beta. Let's get started. So it is the test booklet code is 12, right? If you have that, you can directly check. And otherwise papers, otherwise questions will be jumbled, right? So I am starting with this. I have solved few questions, and I can't even believe paper is damn easy. It is all from PYQs. It is all from PYQs, guys. Right? It is all from PYQs. So you tell me, beta, how are you feeling? Is it good or not? So let's start with the first question. Evil quartet. If you follow my lectures, you know that evil quartet is HOAC. What is this evil quartet? It is HOAC. Habitat loss and fragmentation, overexploitation, alien species invasion, co-extinction. If any of you is my old student, if anyone here is my old student, then he or she, they know it. Right? Right? So HOAC, right? So this is the second answer here. Habitat loss and fragmentation, overexploitation, alien species invasion, co-extinction. So this is what they included. Next question. Which of the following is the site for active ribosomal RNA synthesis? Beta, rRNA. Where will they form? They will be in the nucleolus. That's why nucleolus is also known as the ribosome factory. Why? Because it will synthesize the RNA. That is the question. That is the question. Without any doubt, you can answer that. Next question. In the last class, beta, in the predictor session, we have solved that G1, S, G2 or M. S phase. Very simple hint here. S phase, synthesis phase. Synthesis of DNA occurs, and the amount of DNA per cell will get double, but the number of chromosome will remain same. So B will go with 3. Even if you know this particular thing, you can answer that, beta. You can answer that. As per the biology, I can say it's easy, right? Whatsoever I have analyzed, it's easy to get 350 plus marks in the biology. But let me talk to other educators as well for the physics and chemistry, because I'm not that expert. So I hope then you people, I can then provide you the detailed analysis. And today I will provide it. Don't worry about that. Just make sure you watch our channel. Now, productivity, NPP, GPP, secondary productivity. First of all, GPP, the plants after photosynthesis, what are they going to make? Rate of production of organic matter during photosynthesis. So C will go with 4. And even if I know that, I can eliminate 2 other options. Now, after that, NPP, GPP minus respiration loss. So B will go with 1. Now, after that, even if I talk about the productivity, so productivity will be automatically rate of biomass production, and secondary productivity is what, beta? Secondary productivity is the rate of formation of new organic matter by the consumers. So this is what we have. So second is the correct answer. A will go with 3, B will go with 1, C will go with 4, D will go with secondary productivity will go with 2. Now, next part, you can see here, I was analyzing the paper, even I have solved it, right? Because here we want you to get the accurate solution so that anxiety level should not be there, beta. So whatever we have discussed, it is absolutely correct. So please make sure and do let me know your answers as well, right, beta? Now, Amazon rainforest being cut and cleared for cultivation of soya bean, true that. Now, here they are asking the correct statement. Even if you are confused here, Steller's sea cow and passenger pigeon, they became extinct due to overexploitation by humans. So Steller's sea cow, wait, let me highlight that. And the passenger pigeon, right, they become extinct. Now, third one. Here, what are they asking? They are asking me to choose the correct statement. So, this one, Nile perch introduced into Lake Victoria in East Africa, helped in population growth of cichlid fish in the lake. Obviously not. It destroyed that. So if I have C in any option, I am going to eliminate that option. Now I am confused with D and E. So how to solve that? Water hyacinth is an invasive species. We know that. Terror of Bengal, Ecornia, right, beta? Now, when a species become extinct, the plant and animal species associated with it are not affected. They will be affected. So D is incorrect, right? E is incorrect rather. So A, B, D is going to be my answer. Next part, identify the correct statement about biomolecules. Identify the correct statement about biomolecules. Lipids are generally water soluble. Even if you don't know anything, you know this is incorrect. So I have eliminated the option having A. Now, proteins are polypeptides, true. Polysaccharides are long chains of sugar, true. Adenine and guanine are substituted pyrimidines, they are purines. Almost all enzymes are proteins, true. So D is incorrect. They are asking the correct statement. So wherever D and A were present, I deleted that. I got the second as my correct answer. Right, I got the second one as my correct answer. Now, how many ATP and NADPH? Beta, few days back we have solved it. Right, I think in predictor session also we discussed that. How many ATP and NADPH? But they are saying one molecule of glucose. So we know that one turn of Calvin cycle. In one turn of Calvin cycle, we know that in totality 3 ATP and 2 NADPH are used. So here, because they are asking for the glucose, for one glucose you need 6 turns. So 6 into 3, 2 into 6 into 2. So yes, 18 ATP, 12 NADPH2 will be there. So you can see the answer clearly. Now, there are the following statements which are not true regarding the restriction endonucleases. They are called molecular seizures. Yes, they are even known as chemical scalpels. These are the enzymes responsible for restricting the growth of bacteriophage in E. coli. Absolutely correct. They cut the DNA only at the center, no, a little away from the center. That is why they provide the sticky ends. So C is incorrect. And here, they asked me the incorrect statement. So this is going to be my, this should be in my answer. They remove nucleotides only from the ends of DNA fragments. Beta restriction, endonuclease it is. So end should not be there. Right, anywhere other than, any place other than ends. So C and D. So obviously beta, these two are the incorrect, and yes, they do recognize the palindromic base sequence. You remember for what? For your eco R1 we discussed na? It is Gattus. It is Gattus. Now the next part is, here you can see decomposition, detritus, mineralization or humification. So when you people talk about the decomposition, right, here you people can see breaking down of complex organic matter into inorganic. I told you focus on this organic, inorganic. Inorganic is the thing here, right? Baki still, let's focus and then let's mark it, right? Mistakes should not be there. So I selected it on the basis of detritus, ki dead remains of plant and animal including fecal matter. So if my B is going with fourth, I am directly getting this as the answer. I have no need to check. I have no need to check any other option. Now, ovules are not enclosed by ovary wall, right? And remain. Ovules are not enclosed by ovary wall and remain. So we know that seed formation, where that seed formation will start, we know that in gymnosperms and in angiosperms, beta. In gymnosperms and angiosperms, the seed formation is there. They are the spermatophytes. They are the phanerogams. So in the case of gymnosperms, the seeds are what? The seeds are naked, but here they are covered. That is why we are selecting the pinus here, right? We are selecting the pinus here because on that cone, the ovule is directly there. It is exposed before fertilization, even after fertilization. So it is not covered. It is not covered with the ovary wall. Now, placentation beta, multiple times beta we have solved it. Multiple times we have solved it. If anybody is having doubt now, you check my predictor, right? This NEET replica paper, this is the question that we have solved there as well. So you people can see, I told you exile placentation is my favorite. It will go with 4. B will go with 4 rather. I got my option. I have no need to see any other option as well. Right, beta? So next part, in angiosperm, root hair. Beta, I don't know whose paper it is. So root hair arise from, we know that root hairs will arise from the maturation zone. From where will they arise? They will arise from the maturation zone. Now, which of the following is not a characteristic of plant cells which are in the phase of what? Which are in the phase of elongation. Which are in the phase of what? Which are in the phase of elongation. Yes, everybody? Yes, everybody? So, here you people can see that clearly, right? Then here you people can see that clearly. So which one of the following is not a characteristic of plant cell in the phase of elongation? So elongation is there, vacuolation will increase, cell enlargement is there, new cell wall deposition is there, but large conspicuous nucleus is there in the meristematic zone. So I'm providing the solution as well. So this is what you people can see. Next, which are correct with the reference of transcription point? Last day, the same question we have solved, same thing I have mentioned, beta, same thing I have mentioned, right? This is my transcription unit. Right, this is my transcription unit. Yes or no? So when you look at the transcription unit, so you people can see that very clearly. You people can see that very clearly. Template strand will have the polarity 3' to 5'. So here you have promoter, here you have the terminator. This is my template strand. Now 5' to 3' is there. This is my promoter. Here I have my coding strand. Here I have the terminator. Right. So I have, even in my classes, I always mention when nothing is mentioned, the reference will be made in accordance with the coding strand. So transcription unit is having promoter, structural gene, terminator. It is easy. A is correct. Promoter is said to be located towards the 5' end of the structural gene. So here the reference is made because this is the 5' end in accordance with the coding strand, and that is true, beta. When nothing is mentioned, you will go with the coding strand. Now, the promoter is a DNA sequence that will provide the binding site for the RNA polymerase. Obviously, we have the Tata box here. So promoter defines the template and the coding strand, right? Ki promoter defines, this is the template, this is a... Now see, if you will see, if you are confused in this, ki madam, I am not aware whether this is correct or incorrect. So obviously, beta, look at the options here. Look at the options here. So you have A and your D is included. D is included. So now terminator is located towards the 3' end, right, beta, of the coding strand, and it defines the end of the process of transcription. So out of all the options given here, because one is A, C, D and E only, but here I would like to specify that they are saying promoter is said to be located towards the 5' end of the structural gene. So when no reference is given, beta, they take the reference of the coding strand. So yes, I believe that A should be the correct answer here. So anybody having any doubt here in this particular question? Yes, everybody. Anybody having any doubt here in this particular question, then do let me know. Then do let me know. Yes, everyone. Then do let me know. Done. Next, alpha helix. You remember alpha helical structure, beta pleated secondary structure. Now, which of the following statements are correct regarding the amino acid? Are they substituted methanes? True that. Serine is not, beta, aromatic. We know it very well. You remember in NCERT, the diagram is also given, right? Now, valine is a neutral amino acid, absolutely. Lysine, beta, multiple times we have discussed that it is the basic amino acid. So they are asking the correct statement. So A and C is there. Main function of bulliform cells. Last year also question was there from this part. They are also the motor cells, beta. In the class also we have discussed. So grasses, right, they will, right, it is related to what? We know it very well. We know it very well, beta. So to make the leaf impermeable to fungal spores? No. To perform photosynthesis? No. To minimize the water loss, right? Water loss during water stress? Yes. To transport the water. If you remember, if water stress is not there, motor cells, they will become turgid. The leaf surface will be exposed. If the water will release, they will, like water stress is there, they will become flaccid. The leaf curling will be there. That's what you need to remember. Now, next part. Find the incorrect statement about the photosynthesis. Find the incorrect statement about the photosynthesis from the following. What is splitting complex? Beta, multiple times we have discussed that. Even in the marathon sessions also, it is associated with PS2. So definitely it is incorrect, and they are asking what? Okay, find the incorrect statements, right? So obviously find the incorrect statement. A, C4 plant use C3 pathway of CO2 fixation as main biosynthetic pathway. Yes. C4 plant photorespiration doesn't occur. True that. So it will not be our answer. B and C cannot be our answer. B is given, right? So you can cut that. Moreover, C3 plants, right? So they cannot be because they are correct. So C3 plant, they exhibit Krebs anatomy. No, it is the feature of C4 plants, right? C4 plants. So ATP synthesis in chloroplast occur through chemiosmosis. This is true. So here A and D, because they are asking the incorrect statement. They are asking the incorrect statement. Next question, beta. Conjunctive tissue, again detailed, in detail we have discussed. Beta, conjunctive tissue is there. Just this is your xylem. Let me show you here. Let me show you here. This is your xylem. So in between these xylem, phloem and phloem is there. You know that this parenchymatous tissue is in between is what? It is the conjunctive tissue. What's that, beta? It is the conjunctive tissue, right? So it is the conjunctive tissue. We know it very well. So if we have to figure out the answer, Casparian strip, endodermal cells with suberin deposition. So B will go with 4. Number 1. Subsidiary cell, special cells in the vicinity of guard cell. You remember this particular diagram? Right. Even in our daily practice questions, we included that. Right. Then beta, starch sheath, you know that it is in the stem. Here the endodermis is without any Casparian strip. Starch grains are there. So endodermal cells rich in that. So D will be with 2. So on the basis of that, we got our answer. Now the next one, the simple one. Genetically modified organism. Here beta, I just checked a simple question, that is thermostable DNA polymerase, which I am getting from Thermus aquaticus. So B will go with 3, and I got my answer here. I hope I'm clear. Now, heterophilous development in response to the environment. That's why even if a question is not visible, beta, I am explaining it so that you can see the answer. Okay, so heterophilous development in response to environmental, we know that beta, it is plasticity. So there is no question so far which we didn't discuss in these last few days MCQ practice, beta. It is very easy. Now in race moss inflorescence, race moss, beta, the floral axis will grow continuously, right? The newly found flowers are near the axis, the older one will be at the base. So here, right, as per the option, it is showing the acropetal succession. Now, glutamic acid will be replaced by valine, beta. Sickle cell anemia revised multiple times. Now, incomplete dominance, I told you the examples are important. So, codominance, ABO blood group, yeah, pleiotropy. See, what are they saying? Right, inheritance. Here what I just checked, simply polygenic inheritance, human skin color, right? So D will go with 1, beta. I got my answer. Even if you know the one option, you were able to answer it. So incomplete dominance with this antirenum, right? The codominance part will go with the ABO blood group. AB blood group is the example. Pleiotropy, multiple phenotypic effects of a gene. Phenylketonuria is the example. Again, detailed, discussed in detail. Now arrange the following in accordance with the microsporogenesis. Obviously, beta, because here sporogenous tissue is given. What is given here? Sporogenous tissue is given. So when you talk about the sporogenous tissue, you will have pollen mother cell that will undergo meiosis. After meiosis, we know that the microspore tetrad will be there. The microspore tetrad is going to be there, and then obviously you are going to have the PGs, that is pollen grain. So sporogenous tissue, I have mentioned it, BDAC, sporogenous tissue, pollen mother cell, microspores, diad, then tetrad, and finally that microspore will form the pollen grain. Am I clear? Am I clear, everybody? Sure, bache? Sure. Are you sure? Are you sure? Done, beta? Chaliye, let's go to the next part. Here you people can see, arrange the following steps of DNA fingerprinting. Beta, here, right? Some of you, right, might think that this is, little tricky or something, but it is not like that. Yes, beta. So you can simply see here, right, what I did, simple. So many options are there, right? Finally, we know that, right? Firstly, obviously, what we need to do, isolation of DNA and its digestion. Then we will go for what? we will separate the fragments. We have to separate the fragments. Now, transferring of these separated DNA fragments to the synthetic membrane, and then we are saying hybridization will be there. After hybridization, detection of the hybridized DNA. So, even if you don't know, you know one thing, hybridization, after hybridization only you can just detect. After hybridization only you can detect. So, I just checked this, right? That B, a hybridization, and then D. So BD in a sequence, they are given in this option only. That's how I opted this as the correct answer. Is that clear? Is that clear? Is that clear? Sure? Done, beta? Any doubt? Any doubt? Chaliye next. Exploring molecular, genetic and species level diversity for the products, beta, for economic importance. These terms I have even posted in the shorts so that students can get to know. This is bioprospecting. Bioprospecting, you are using the living being so that you can get the economic benefits. So this is bioprospecting. Now, after this, right, so bioprospecting, which of the following statement are true with reference to the sex determination in honeybees? We know that they show haplodiploidy. What do they show? Haplodiploidy. Now, an offspring formed from the union of a sperm and the egg. Now, asking, what are they asking? True statement, right? See, I checked what? Out of all these, what I checked? Male produce sperm by meiosis. No. Why? Because male here is what? Right. Male here is what? Male here is haploid. So because male here is haploid, right? So we know that when a haploid gamete, it is the parthenogenesis, because this haploid gamete is directly growing into an adult, and here it is the male. So a henotoxy, this is what we define, but okay, you don't need the technicalities here. So D is incorrect, right? And they asked the true statement. So because they asked the true statement, so wherever we have the D, we incorrected that and we got the fourth as the correct answer. Now, correct sequence, my favorite trick, die. Denaturation, annealing, extension. Denaturation, annealing, extension. Denaturation, annealing, extension. So which of the following statement are correct with reference to the, with respect to the DNA separation? Now, these are the questions which we will solve here and we are going to discuss that. But up to this part, tell me, up to this part, any doubt? Up to this part, any doubt? Yes, everybody, up to this part, any doubt? Any doubt do you have? So again, they are saying which of the following statements are correct with respect to the DNA separation, isolation and visualization. First is the DNA separation, then you have to isolate and visualize that. They are asking the correct answer. Cutting of DNA is done by molecular scissors, that's true. DNA fragments, they separate according to their size in agarose gel upon electrophoresis, true. Separated DNA segments can be seen without staining. No, beta. Stain with ethidium bromide is important. So I will eliminate this option here. Now, separated DNA fragments, when stained, they are seen in visible light. Again, this is a direct MCQ which we have solved in our previous sessions, in our predictor session also. So here it has to be UV light. So obviously we know that D is also incorrect. So we are left with the fourth option. We are left with what? We are left with the fourth option. Now, main criteria for 5 kingdom classification, beta, as such, it is 1969. It is as such we have discussed. Do we have cell structure, body organization, reproduction, phylogenetic relationship, but presence or absence of this flagellum, it is not something that we have to consider. So I eliminated 2 options. So reproduction part was also there, phylogenetic relationship was also there, right, beta? So see, ABE, common to both, it is correct. Now what about reproduction? They considered that, yes, beta, they also considered the reproduction aspect also, right, beta? But the main criteria was what? But the main criteria was basically that they focused on the mode of nutrition. Is that clear? They focused on what? They focused mainly on the mode of nutrition, right? That is the important thing. Just allow me 1 minute for this question. So here, right, just 1 second, beta. So ABE we have, ABDE we have. So what do you think? What should be there? What should be there? Do you think that they considered the reproduction part or not? Right, they considered the reproduction as the main cause or not? Anybody here? Anybody here? Yes? Anybody here? Right, let's reconsider it. So this is to for sure because this is common to all and C is never there. So I have eliminated this option, this option. Now in such options, yes, we might get confused. Whether they have considered the aspect of this reproduction or not. So you tell me what should be there. Yes, everybody, you tell me what should be there. And meanwhile, Gaurav sir is also ready, right? Because we believe that you should know, right? Parallelly, you should know all the three subjects, their solutions, so that even you should not feel bored, right? So tell me if there is any doubt here. Yes, everyone. Quick. Yes, anybody having any doubt? So do we have the reproduction part here also? Yes, they considered the reproduction part here also. So Gaurav sir is also ready with the chemistry solutions, right? So we will discuss few more questions from this part, then Gaurav sir will come up so that there should be a balance. You should feel like it, okay, bio is going well, chemistry is also going well. So which one is the triploid cell? Which one is the triploid cell? Obviously, beta, without any doubt. This time to NTA is like, take the previous year question. Take all the previous year question. Chaliye. Next part, which of the following statements are correct with reference to the packaging of DNA helix? Histones are organized to form a unit of 8 molecule called histone octamer. Correct statement they are asking. Histones are negatively charged basic proteins. No, beta. B is incorrect. So I will eliminate these two options directly. Histones are rich in lysine and arginine, true that. Positively charged DNA is wrapped around the histone octamer to form nucleus, true that. The packaging of chromatin at higher level requires an additional set of protein called non-histone chromosomal protein. It is also true. Now let's see the options here. Right, B is incorrect. Right, B is incorrect. Now they are saying histones are organized to form a unit of 8 molecules called histone octamer. Beta, octamer to we have two copies are there, right? Two copies are there. Now, what do we need to see? We have A, okay, done, beta. We have A, or in the answers we have ACE, we have CDE. Okay, so ACE, CDE. So histones are rich in the basic amino acid residues, right? Obviously, what do we have? Yes, lysine and arginine, true that. Positively charged DNA is wrapped around the histone octamer to form the nucleosome. That is also true. Packaging of chromatin at higher levels require an additional set of protein called non-histone chromosomal protein. That is also true, right? So basically, out of all that options, now we are confused with the A and D. So D is the positively charged DNA. Beta, D is incorrect. Yes. D is incorrect. DNA is not positively charged. DNA is negatively charged. Very good. So DNA is negatively charged. So D will not be the option here. So second is the correct answer. It is going to be ACE because yes, beta, DNA is the negatively charged. They have mentioned it positively. Now, which one of the following is in situ conservation? What is in situ? In situ is the on-site conservation, right? On-site conservation, right? In their habitat itself, the organisms will be conserved here, beta. So wildlife safari to definitely not there. So they are saying which is of the following is an in situ conservation method. So obviously wildlife safari is not there, botanical garden, seed bank not. So it is going to be the sacred groves. That is our answer. So in lac operon, again the one that we have solved, the Z gene codes for what? This one is going to code for, because it is ZYA, Z will go for beta galactosidase. Very simple. Now here again the hormones part, beta, hormones part here. So stomata closure, right? D will go with 2. So we will eliminate these two options, right? 2 for D, dichlorophenoxyacetic acid. It is A will go with what? A will go with 3. So this is my answer. That's it. Now arrange the following steps of somatic hybridization in a correct sequence. Now somatic hybridization is there. So first of all, beta, D will be there. Why? Because you have to isolate the single cell from two different varieties. Then you will go for their digestion of cell wall. Then you will obviously isolate the naked protoplast. So DAB. Do we have DAB anywhere? No. So next is digestion, then isolation. After that, fusion of protoplast to get the hybrid C, and then we have the E. So DABCE is going to be the correct answer here. Is that clear? And beta, this question as such we have solved. all the paper is basically from the p-values. This question, if you remember, as such we have solved, even if you don't know that, so you know that what has to be there, right? You know that it is basically from the RQ value, right? So volume of, right, what, how will you check it? You know it very well. So tell me what you marked as, so you can clearly solve it as well, and you can get to know the answer. Is that clear? So basically, before proceeding here, right, RQ value, we know that, beta, right? So what is the RQ value? I hope you all are aware of it, right? The same question we have solved, you have used more oxygen and less CO2 is released here. So obviously we are talking about the fats here, beta. We are definitely talking about the fats here. Is that clear? So here the student has marked the answer incorrectly. So you people should know what should be the correct answer here. So now Gaurav sir will come up, right? And then I will come up with the biology part, because we are here to make sure, right? We are here to make sure that you get the accurate answers in less time. Is that clear? Done, beta? Chaliye. So meanwhile we will solve, and Gaurav sir is ready here. Okay. Okay. So this is the simple calculation that you have to make, right? This is a simple calculation that you all have to make, and trust me, the paper this time, right? The paper this time was very, very, very easy, isn't it? Isn't it? Right, beta? So, isn't it? So here this time you don't have the direct answer for this. Okay? Okay, so let me ask the Gaurav sir if he's ready. Okay, so he's ready. You solve chemistry. Okay. Done? Otherwise, I'll continue. I think, sir.
Yes, guys. So I hope you had an awesome discussion of biology. Let me just set this brightness properly. Now, how was your paper, guys? How was your paper, guys? How was your paper, guys? Chalo. Let us move forward. Beta, now, see, this is what a calm mind can turn your paper into. If you just really try and see that what is the level of question paper, then you obviously have to attempt those questions with a very calm mind. Now, why I'm saying so, because see, once now we have got the structured questions. So once you have these structured questions, now once your mind is calm, that paper, if you have a blur paper, the blurriness will obviously create a chaos. Okay, so now let us start, sir. Now this is very doable, because this is very clean rather. So you just try and find out that whether they are asking anything out of NCERT or very easy or not. This is definitely, sir, cumene. So once I have cumene, sir, this is isopropyl benzene. We already discussed it, but yes, all the options were not clearly visible to us, but now I guess they are. So if I have cumene, oxidation of cumene will do what? Sir, oxidation of cumene will oxidize the benzylic carbon. So where do I have my benzylic carbon? So this one. So this H rather will become C bond OH, and remaining two methyl groups are definitely as it is. And after that, hydrolysis. What hydrolysis, sir? Acidic hydrolysis will turn it into phenol. And what will be the byproduct, sir? Sir, byproduct will be acetone. So this basic production of phenol was actually asked, which we have clearly seen also. So that means this is 2. So option 1 has been cancelled. Now, carboxylic acid has to be turned into an alcohol. So what will be the option for this, guys? If you just see, sir, oleum acid, we do not find, but with alcohol and acid, so any carboxylic acid with alcohol, rather, because they have used simply methanol in acidic medium, it will form an ester. Sir, the methyl ester of ethanoic acid, so this will rather be methyl ester of this ethanoic acid. Now it is subjected to reduction. It is clearly mentioned in carboxylic chapter of NCERT that if you need to form alcohol using expensive reagents like lithium aluminum hydrides, the alcohol will not be sold to that price. So what is the cheaper version to do that? Catalytic hydrogenation. So this catalytic hydrogenation will reduce the will give you the 1 carbon alcohol, that is methanol is generated back, and you will be getting the 2 carbon alcohol. But yes, to this I would be considering as my major product because that I have already been used. So this is ethanol. That means B is clearly going with third. Now that means I have eliminated one more option. But yes, you have to be vigilant enough to attempt such questions. Okay guys, chalo. Now after that, sir, this is 3 carbon alcohol and it is getting converted to this is propane 1-ol getting converted to propane 2-ol. Now what to do over here, sir? Sir, it is getting reacted with concentrated sulfuric acid. What does concentrated sulfuric acid do? It do dehydration. So first this alcohol will undergo dehydration. Dehydration is going to give you alkene. What alkenes are? Prop-1-ene. Only one product is possible. Now that prop-1-ene will undergo reaction with H plus in water in acidic medium. So that is acidic hydration. So at acidic hydration of prop-1-ene is going to form Markovnikov hydrogen OH added product, that is propane 2-ol. So your answer will be propane 2-ol, sir. That means the isomers are produced. This is because this is nothing but propane 2-ol. So that means C goes with 4. So I guess if you just do C goes with 4, you have definitely got your answer. Answer to this question is definitely D. For that, you do not even have to attempt the option D. But yes, because we are practicing, yes. Sir ji, mere भैया ने एग्जाम दिया था. ओके बेटा, आई कैन नॉट स्पीक इन हिंदी because just read the channel name. This is PW NEET English, so we are supposed to speak in English, beta. Acha, by the way, we have not explored option D because we have clearly got our answer, but still because we are practicing, paper is done. I am here to discuss this to all the students which are going to give paper in the later examinations. So all NCERT facts. Now benzene converting to alcohol, sir, we are only left with one option. So actually benzene with oleum, I'll give you the detail also. Benzene with oleum, once I have benzene, what is oleum, sir? H2S2O7. It will give you benzene sulfonic acid, that is SO3H. Now remember one thing, if although they have not asked, but a very, very beautiful question is, what is the actual electrophile? Electrophile is a neutral SO3. So this will form benzene sulfonic acid, which becomes rather a very good leaving group. So once it is subjected to base and heating followed by hydrolysis, it will give you the required aromatic alcohol, that is phenol. So definitely it will form the corresponding phenol. But yes, for an examination you do not even have to attempt this because till option C you have eliminated all the options. Moving to the next one, I hope it is very clear to everybody. Okay, I am dropper any batch. Naveen, yes, obviously we have, for dropper we have Yaqeen, but yes, definitely it is in English. But I hope it will help you. Yeah, the paper code is 12, beta. The paper code is 12. Chalo. Moving to the next one. The major product Z formed in the reaction sequence. Now this is our next question where we have to give what is the major product. So, sir, what is this? C2H6, that means I have CH3 bond. CH3, that means I have ethane. Ethane on reaction with Cl2. Now what does Cl2 do? Cl2 in presence of sunlight. Sir, this is free radical substitution. So hydrogen will be gone, a radical will be produced, and chain termination will give you a monochloro product, which is nothing but a chloro derivative of ethane. Now with ammonia, sir, again it is very easy. Why? Because this is simple SN reaction. This is minus, this is plus, lone pair will go, and you are beautifully going to get your 1 degree amine. A very easy question. Now the previous question also was very doable, but yes, it was lengthy because you have to go through 3 reactions, because yes, you can leave the 4th reaction because anyway with 3 you were getting answer. But yes, with such questions it is very easy. So that question I would rather say was a moderate one, but this is an easy one. So ammonia will give I've got my 1 degree amine, which is definitely, now why? Now, this Y is treated with NaNO2 and HCl. We definitely know nitrous acid is unstable. It is unstable acid. So because it is unstable, so we have to create it. So this nitrous acid is produced by a mixture of NaNO2 and HCl, followed by hydrolysis will bring about the reaction of first diazonium salt will be produced, nitrogen gas will be released, and you will be getting your corresponding alcohol. So your answer will definitely be option Bombay, right? Chalo. Next, after that, let us move to now. Acha, please tell 6:30 beta. Yes, once we are done with the discussion of the paper, then definitely we'll be beta. First let us discuss the questions now, because anyway you have submitted your OMR sheet, so whatever answer you have got will be mechanism cumene to phenol is explained is okay. What, Arvind, what do you find wrong in that? Beta, I didn't even give you the mechanism. I just gave you the NCRT flow. The mechanism, I haven't. For a NEET level of examination, you do not even have to attempt those mechanism. I have given you the NCRT flow. That was not a mechanism, beta. Chalo. Moving to the question from ionic equilibrium. A very simple, rather very simple question from sparingly soluble salts, because value of K is given, which is 10 raised to the power minus 10. That means it is not at all an electrolyte, but still, let's say X amount will be dissociated. So let's say X amount of this is dissociated. So X moles per liter and X moles per liter of these two are formed. So, but what never changes, value of K, and value of K is definitely X square in this case. What is the value of K that is given to us? Equilibrium constant is given out to be 10 raised to the power minus 10. This is value of X. So X will be under root of this. So what shall be its partial solubility or partial ionization in water? Sir, it will be 2 into 10 raised to the power minus 5. I hope everybody is clear with me. I hope everybody is clear. Chalo. Beta, if X is this, then that is the concentration of OH minus ion as well. So what will be POH? Sir, minus of log of 2 into 10 raised to the power minus 5. So minus of log of this is actually minus of log of 2 and minus of log of 10 raised to the power minus 7. What else, sir? Minus of log of 2 is nothing but 0.3, but with a negative sign. Although I guess log value they are giving also, because they are answering you till 3 places of decimal. So, but yes, I guess we can approximate. And minus plus 5, because log of a raised power b, and log 10 is definitely 1. So this is 5 minus 0.3, which is nothing but approximately 4.7. Why I'm saying approximately? Because they are saying to calculate till 3 places of decimal. So why require? Let us first find an option, and then we see that whether we have to do that calculation so precisely or not. So value of pH will be nothing, 14 minus POH, because in the question you are supposed to calculate value of pH. So pH will be what, beta? 14 minus this, that means somewhere around 9.3. So which option I have somewhere around 9.3? Option C. So why we should do that calculation unnecessarily? I hope, yeah, beta code is 12. Code for this paper is 12. Good. Chalo. I hope we are done. Chalo. Moving to the next one. Yeah. Now, next question for code 12 paper was code 12 beta, not 13. This is code 12 paper we are discussing. Hana? Chalo. So we are discussing. I mean, I am not even bothered with the number of now question number. I am bothered with the concept. And yes, you should know the concept, and obviously because you have given your examination, so in one or the other form you may have got this question. So let us. Sir, physics का paper कैसा बेटा? Just after we will be keep switching so that whatever questions we are giving to you, they have been cross-verified and rectified, because we want to give you whatever is the best. So yes, right now I'm discussing chemistry. Then obviously after this, maybe your biology ma'am or physics sir will be definitely joining. Shubhrat sir and Ambika ma'am will definitely be joining. But yes. सर 8 साल से मेहनत कर रहा था, अब की बार PW ऑनलाइन से हो गया लगता है 647. very, very, very best of luck, Vipul. Congratulations, beta. All power to you. This is because of your hard work. Congratulations. Many congratulations to you. Chalo, beta. Congratulations. Chalo. A very good score, I would rather say. 647 is a very good score. Deuce, beta, we'll soon be discussing the cutoff as well. But yes, initially I'm more interested in discussing the questions with you, and then obviously we'll move forward for the. Nahi, beta. The paper is the chemistry questions which I have done right now. I would rather say one was moderate, remaining were easy, and I guess we have finished like 3-4 questions right now. है ना? Chalo, we are in no hurry. Yes, sir. Sir, beta, Mr. Momilin, the reason I'm discussing this is because after you, na beta, have that compassion, there are more juniors of yours, or maybe if God forbid, if you also have to attempt the NEET examination next year, then these analysis actually gives you a kickstart, because you get to know, okay, this is coming, okay, NCRT is so important. So taking time is actually the job right now, because beta, in that way you will feel the connect. In that way you will feel actually this is the problem. Paper is very easy, beta. Whatever chaos we have made is around the process. Yes, because the process has become tedious, because there are so many students attempting and going for a thing which has 2% of available seat, but 100 people are approaching. So this is just that chaos. That's it. Chalo, beta. Now let us move to the question. 1 liter, because decimeter cube is 1 liter. So 1 liter of carbon dioxide gas is passed over red hot coke. Okay, sir. That means CO2 gas is passed over carbon solid. Now, obviously, if you heat them, question is saying that 1 liter, after the reaction, the volume is becoming 1.4 liter. Sir, volume is increasing. That means I need to form a gas. You think of your inorganic side of chemistry, what it will be. Sir, carbon taken by CO will form the poisonous carbon monoxide. This is the only inorganic knowledge which you should need. You can predict this also, because volume is increasing. That means more gas should be formed. And chemically, how will they react? Sir, if you increase carbon, then number of percentage of oxygen is decreasing. So you get a product which is not completely oxidized. So what will be your product? Carbon monoxide. Now, if we balance the reaction, carbon is 1, carbon is 1. So that is also 1. Oxygen. Okay, but that is 1. So if I multiply a 2, so I guess this is a balanced reaction. Now, after that, what we have to do? Sir, just do the stoichiometry. 1 mole of this reacts with 1 mole of this to produce 2 mole of this. I had 1 liter of this. Let's say X liter of this has reacted. So X of this will also be reused. But why bother? Because this is a solid. We are only interested in volume of gases. And obviously 2x liters of the product will be produced. Everybody is good with it. Now, total volume of gas after reaction will be what? Sir, this plus this, that means 1 minus X, that is of CO2, because this is the remaining gas, and 2X will be of carbon monoxide, the gas produced. Now that means this is nothing but 1 plus X. I guess value of 1 plus X is given to us, which is 1.4. That means X is nothing but 0.4. Now, sir, this is quite a piece of cake for us now. You have to give the composition of gaseous mixture. What gas I left with? Sir, CO2. So CO2 will be 1 minus X, as in 1 minus 0.4, as in CO2 will be 0.6. So CO2 will be 0.6. No, yes, no. So option C is your right answer. You do not even have to calculate for carbon monoxide. I hope everybody is clear with this. Sir, I am in class 10th, Kohinoor 2.0. Okay, beta. Mir Momilin. Okay, beta. Chalo. I hope we can move forward. Sir, is paper lengthy? Haan, beta. I would rather say, if you have invested sufficient time in your practice, then it will be quite doable, or else paper is lengthy. Paper is not, yes, it will take time, it will take efforts. So yes, common sense will be required, and your accuracy, once you are practicing more, will be required. So I would not say that paper is very easy, because we are time restricted also. We have to attempt these 45 questions in a limited time. So yes, it is lengthy. Chalo. Now, this is a very, very easy question, beta, from quantum theory. N is equal to 2, L equal to 1. L is 0 for, sir, L is 0 for S subshell. L is 1 for P subshell. L is 2 for D subshell. L is 3 for F subshell. So this is nothing but 2P. This is nothing but 4S. This is nothing but 5F. This is nothing but 3D. Just compare. So this is 2P. That means A is 2. That means A is 2. So these options are gone. Then B is 4S. So this is 3. Now you do not even, acha, okay, this is also there. So we again have to check. C. C is 4. C is 4. Huh? So C. Answer is option A. I hope everybody is clear. Guys. Chalo, perfect. Moving to the next one. Again, a very traditional question from hydrocarbons. A very traditional question from hydrocarbons. Let us see what the question is asking us. The number of chlorine atoms present in organic product X and Y. Again, the number of chlorine atoms present in product X and Y. This is X and this is Y. Sir, traditional question. Yes, how traditional, beta? Benzene reacting with chlorine. Anhydrous AlCl3. Sir, this is Friedel. This is anhydrous AL3 is actually, but we rather do not use the word Friedel Crab because we use for alkylation and acylation. But yes, Friedel Crab, this Lewis acid is there, which will definitely break chlorine into Cl minus and Cl plus. So the electrophile that will be created, because excess of electrophile be created, so they'll be doing excess of electrophilic substitution. So what are you going to produce, sir? Perchlorobenzene. Every hydrogen will be substituted and I will be getting perchlorobenzene. No hydrogen, but benzene is still there. That means this is nothing but C6Cl6. So how many chlorines you have? 6, sir. So you are gone. These two may be your answer. Now, what about Y, sir? Sir, now this is sunlight. Sir, providing sunlight to benzene is a vigorous thing. You are not supposed to do it. But if you are doing it, then aromaticity is gonna die. So this is an addition. Hydrogen will be remainder and chlorine will be added. So this will be CH all the way, because aromaticity of benzene ring is gone big time. And I am introducing chlorine all over. So answer for me will be C6H6Cl6. But although this is not benzene, but because it is industrially very useful, so it is called benzene hexachloride, also known as gemexane. And how many chlorine? They are not asking any of those things. They are just asking how many chlorine. See, so how many chlorine? 6. A very rather doable question, guys, right? I hope everybody is good. Good. Okay, I see C over there. Now we are going on the yesterday's pace. Now everybody's attending this analysis with a motive of learning. You know why I'm feeling this? Because now people have started answering also, sir. See, that means now they are in reading mode again. Because yes, all of those who have given your examination, this is your day, beta. Just hold on, see the results, compare with your paper what you have done, and yes, after that you should definitely take a break. You should take the day off, go out with your family. But yes, first let us compare the answers and have that level of compassion. The reason I'm discussing everything in detail, because I want my class, your juniors, to have that compassion now and start being religious and disciplined with their content. Because yes, beta, paper is very much doable. You need more readings ability. So your ability to read words per minute should be very sound. This is the only, I guess, the only thing, the main requirement from a teacher's side for a student to excel in the examination is your reading speed should be super high. है ना? चलो, हाँ, next. next. I am not giving you so much of ज्ञान. चलो, moving next. I understand. चलो. In the following reaction sequence, X, Z. Okay, you have to give me X and you have to give me Z. Okay, sir. What about X, sir? So this is 3-carbon alcohol reacting with PCl5, giving you obviously the corresponding alkyl halide. And what will be X, sir? Because this is PCl5, so with PCl5 you form what, sir? You form POCl3, phosphorus oxychloride. I hope everybody remember this. Hello, hello. Chalo. I have failed. Beta, nobody. No, don't say that. If you still have got the remainder amount of options in your life, you just if at all you're feeling so, then it's just a phase, it'll pass. But yes, at least recollect or yourself, reorganize yourself and aim for better in your life by working hard, है ना? Chalo. So this is POCL3. So X is definitely POCL3. So this can be my answer. No, you are gone. No, you are gone. Option D is also POCL3. I hope you can see that. Now, this alkyl halide is reacting with alcoholic KOH. Obviously, alcoholic KOH will bring about beta elimination, and beta elimination will bring you an alkene. So this is why, but the question is not asking you why. So this is my alkene. Now this alkene is subjected to HBr, but in presence of peroxide. So in presence of peroxide, HBr do what? I'm sorry. In presence of peroxide, HBr do what, sir? It do anti-Markovnikov addition. So anti-Markovnikov addition of HBr will produce what, sir? Sir, it will produce 1 bromo. Why? Because here the free radical will be formed here, and once bromine has attacked, and after that this will attain that H. So your answer will be 1 bromo, and where you can find that 1 bromo. So option 4 will be your right answer. I hope everybody is good with it. Kitna drop? Acha, people. Okay, I hope it is good, guys. Okay. Timing of beta we'll be switching. I mean, after chemistry, Ambika ma'am or Shubhrat sir will be joining. I'll just confirm, but yes, at least let me give you how many questions I have. I don't have. Acha, okay. So the remaining questions are still getting typed. So let's see if your next faculty is available for you, or maybe I have got few more questions. So let's see. Nahi. Chalo. So by the time let us attempt few more questions. Maybe few more questions for me have got. Let me check. I have few more questions for you, because see beta, giving you those questions from paper was not working. It was not working properly. Chalo. So yes, I think now I have got few questions, and let me try out those questions for you. Okay, yes, I have got few questions. So let us try those questions. I have got those questions printed for you. Now see the level and commitment we have and we keep in order to give you whatever the best we have with us. So we have the paper we have just collected and got that typed. Okay, so I guess chalo beta. So we discussed this. I guess this we discussed. This we have to discuss. Okay, chalo beta. Chalo. Let us attempt this one. Guys. Now the question says V2O5. Sir, V2O5 is vanadium pentoxide. Vanadium pentoxide is used where? Preparation of ammonia, polymerization of alkynes, preparation of H2SO4 from SO2, preparation of H2SO4 as in sulfuric acid. Sir, yes. Is it a block chemistry logic? Yes, beta. It is a block chemistry logic. But because, but just let us explore the questions. Oxidation of ethyne to ethanol. So let us first write the reactions, and then, sir, please discuss fast. Beta, see, let us write the reactions and we'll predict what will be the reagents. Preparation of ammonia. So hydrogen reacting with nitrogen forming ammonia. So this is Haber's process. Polymerization of alkynes. So if I have any alkyne, let's say the simplest alkyne, you can have cyclic polymerization or you can have linear polymerization. So that I hope clicks us. So let us now compare that preparation of ammonia, which is one, is obviously Fe. This is obviously Fe. Yes, promoters are used, but what is the catalyst used? Sir, catalyst is definitely Fe. So if that is Fe, then B goes with 1. So B goes with 1, B goes with 1. This one is cancelled. Now after that, sir, A, which is V2O5. Then I have C, which is PdCl2. So PdCl2 and nickel complex. Nickel complex, I guess. So there is, I guess, an option problem, na? There is, I guess, an option problem beta over here. Let me just confirm this, because 4. Nahi, I will just come back to this question, beta. I will just come back to this question by the time we attempt this one. This is a very basic question from chemical bonding. Identify the correct statement about ClF3. So what about ClF3? Sir, chlorine has got 3 bond pairs, and after 3 bond pairs, it will be left with, after 3 bond pairs, it will be left with 2 lone pairs. So sir, bond pair is 3, lone pair is 2. That means steric number is 5. So obviously it is AB5, but because of 2 lone pair, what shall be the shape? Sir, shape will be, lone pair will be coming in equatorial position, so shape will be bent T shape. So it will have a bent T shape. So let us explore the options. It has a bent T shape. So this is planar, this is trigonal, bent T geometry, and definitely with 2 lone pairs. So B is my answer. A very easy question. Yeah, a very easy question. Okay, perfect. Chalo. Next one. Calculate EMF. Sir, I have discussed the same question. I have discussed the same questions in the lives that we have done. The same question, sir. This is a reaction where hydrogen from 0 oxidation state is going to +1 oxidation state. So how to write the half reaction, sir? Sir, because this is oxidation half, so hydrogen giving you twice of H plus 2 electron. So the electrode potential will be, standard electrode potential minus 0.059 upon number of electron transferred is 2 into log of H plus ion concentration squared, because this is the product. I hope everybody can see that now. Sir, because this is reference electrode, so this is definitely 0. So my answer will be minus 0.059, or else yes, I can approx also, because options are quite far. Let us take 0.06. So this will become 0.03 into 2 into log of H plus. Sir, what do I have? I have H plus ion concentration, which is 2 into 10 is power minus 2. So this will be log of 2 into 10 raised to the power minus 2. Now that's it. The basic calculation now we have to do. So it will be minus 0.06 into, what about log, sir? Sir, it will be minus 2 and plus log 2, which is 0.3. So this is nothing but minus 1. So minus will become plus. So this is 0.06 into 2 and 0.3. So this is 1.7. So I guess calculation is also quite doable. Then this is 7 into 6. 6 is 42. That means 2. So I give a 4. 6, 1, 6 is 10. So this is 0.1 something. So your answer will be, calculate EMF of the half cell, 0.10 something. So your answer will be option A. Right? Because they are asking you, this is the oxidation potential. I guess this is negative, and because I am writing the oxidation half reaction, here also they have, and nowhere they are asking calculate EMF of the half cell. This is the half cell, which is the oxidation half. This is the half cell, which is the oxidation half. I hope everybody is getting it. Now, let me just see that whether I'll be coming back with, okay, so your biology madam is ready with the next questions. I'll be coming back with the discussion of next questions. But yes, by the time you just go and check your biology questions, and then I'll be coming back after arranging these chemistry questions again. Okay, so ending from my side, ma'am, over to you. Over to you.
Done? Okay. So guys, let's resume the paper discussion. Let's resume the paper discussion. Now I will finish it in one go, right? So we are done with the biology part and you are going to get it errorless, okay? Right? So I think we solved it up to which part? Last question that we discussed, it was, yes, this one. This one. So this part again, as I told you that we have also discussed it in our regular sessions also. So here you can see what do you have to see, right? It is regarding the RQ value. This is what I discussed last. So you know that it is volume of oxygen evolved to the volume of CO2. The volume of CO2 evolved to the volume of oxygen used and volume of CO2 evolved. This is what volume of CO2 evolved to the oxygen use. So this is what we check it, and here the value is going to be 0.7. So I told you this, right? By looking at this, you can simply see that oxygen is more, carbon dioxide is less. Like oxygen used is more, carbon dioxide used is less. So this is simply the example of fat. Smart work, 0.7, no need to even calculate. So this is what we are going to mark. Now let's move to the next part, beta. Since the origin and diversification of life on earth, I hope questions are clearly visible. Let me confirm once. Questions are clearly visible, right? So here you people can see, since the origin and diversification on life on earth, there have been 5 episodes of mass extinction of species. How's the 6th one is, which is in progress, is different from other? Beta, it is basically due to the anthropogenic activity, due to the human activity. So the present species extinction rate is 100 to 1000 times faster than in the pre-human times, right? So this is what, as far as lower than those, and no. So this is what you need to know. Next question beta. The answer key here is errorless, so please, right, focus here. So you can see trypsin, again solved one from the biomolecules part. Trypsin will go with enzyme. So A will go with 3 here, beta. Morphine is the analgesic. Multiple times we have solved that. It is an alkaloid. Concanavalin, they are the lectins, and collagen is the intercellular ground substance. Now, which one of the following is not true about the universal rules of binomial nomenclature? So here we know that, beta, the first is going to be the genus or the generic name, and this is going to be the species name, that is specific epithet, right, beta? So both the words in a biological name, when handwritten, are separately underlined. True that. If you are printing it, they should be in italics. Specific epithet in the biological name starts with a small letter. True that. First word here represents the specific epithet, while the second, no, it is opposite. First should be genus or the generic name, second should be species or the specific epithet. So biological name are written in Latin. True. So here they have asked the incorrect statement. So the enzyme required for carboxylation in, that is addition of CO2 in the case of Calvin cycle, which is C3. So you know that we will have the rubisco. So this is rubisco, right? Ribulose bisphosphate carboxylase oxygenase. Now, which of the following formula regarding Solanaceae, though Solanaceae family is not the part of your syllabus, right? Yes, you people can consider that it is outside the syllabus, but obviously you are not going to get any grace or something, right? But Solanaceae family is actually not the part of your syllabus. But out of all the options given here, if you remember, right, today morning also we discussed that part regarding the Solanaceae family. So this is what you have to remember. Yes, but in their case, this some members, they show the epipetalous condition, right? Some members indeed they show the epipetalous condition, beta, right? Indeed they show the epipetalous condition. So how is it different here? Because here they are showing that sepals are also fused. So this is what we have to opt. It is going to be the one. And even if you will go as per the old NCERT, here I would like to specify this thing here, right? No doubt in general, this is what we see in most of the members here, but beta in old NCERT, right, they have mentioned the answer as C also. Let me tell you this thing, okay, right? In general formula is this, but here you can go for that. As of now, you can rely on it. We'll wait for the NTS answer as well. Now, which of the following type of pollination brings genetically different types of pollen grains to the stigma? Obviously, cross-pollination, right? So when it comes to the cross-pollination, which example do we have? We have the example of xenogamy. Is that clear? Now, the next question here is in front of you, right? Everybody can see that. Next question here is in front of you. So you people can clearly see that. You people can clearly see glycolysis, which is basically Ambeden-Meyerhof Parnas pathway. Where will it occur? In the cytoplasm. It is an oxygen independent process, right? ETS, electron transport chain, inner mitochondrial membrane. So here accumulation will be there in intermembrane space. So Krebs cycle, it is in mitochondrial matrix. So A will go with 3, B is going with 1, C is going with 4, D is going with 2. Next part, this question might be, I think it's blurry. Let me read it out. So insertion of foreign DNA at BAMH1. So this is the example of insertional inactivation. It is in the cloning vector PBR322. It results in the loss of antibiotic resistance towards. So we know that it is present in the tetracycline gene. So if we are using this particular, right, tetracycline resistant gene is there. So if you are using this particular antibiotic, so it will result in the inactivation of this tetracycline resistant gene. Is that clear? Is that clear? So this is going to be my correct answer. Next part, 6th mutant code of beta globin chain cause polymerization of hemoglobin and change in RBC. So you know that my favorite trick, GAG will become GUG. So GAG is glutamic acid, right? GAG is glutamic acid, and here GUG is going to be what? GUG is going to be valine. GUG is going to be valine, so that's why it is the correct answer. Next part, beta. Choose the correct statement regarding gift, right? Gift is basically what? Gamete intrafallopian transfer. So the gametes will be transferred, right? Gametes will be transferred intrafallopian, within fallopian tube. Gametes will be transferred within the fallopian tube. So ova collected from female donor transferred to the uterus? No, beta. Clearly in the question, gift intrafallopian, it is a transfer of an ovum collected from the donor into the fallopian tube of another female who can't produce ovum but can provide suitable environment. So early embryo, no, this is gift, right? It is in vivo fertilization. Now sexual deceit, you know that pseudocopulation. So pseudocopulation is where? Ophirus and bumblebee, where the male, this ophirus, which is an orchid, its petal appears like a female bumblebee. Male will think that that's female, but actually it is basically the petal. And with the time, due to evolution, even the flower also start producing the, I would say the fragrance is just like the female part. So this is what you have to remember. Next part, evolution of human appears. Beta, here I literally want to show off, right? I literally want to show off. I repeat this trick multiple times. Dara H2HE-NFS. Dara H2HE-NFS. Even in our last marathons also we discussed. So Dryopithecus rama australo, Homo habilis, Homo erectus, Homo sapiens neanderthalensis, Homo sapiens fossilis, Homo sapiens sapiens. So as per the sequence, beta, so Ramapithecus should be first. Then you can see the order Homo habilis, Homo erectus, Neanderthalensis, Homo sapiens is there. Now, match list 1 with list 2 related to embryonic development at various months of pregnancy. So you can say that fetus movement start, and here appears on the head 20th week of pregnancy, right beta? 20th week of pregnancy, right beta? Now the fetus develops limbs and digits. 8th week of pregnancy, fetus develop external genitalia. Here you can see the 12th week of pregnancy, and body is covered with fine hair. It will be the 24th week of pregnancy. So it is it is not in the trimester form. In the week format, they have mentioned it. Now the next part, beta, a group of researcher. See, this is a very good question. The group of researcher, they procured some fish-like animals, and upon investigation, beta, endoskeleton cartilage. Okay, that can be chondrichthyes also, but this is ectoparasitic. They are found attached on the fish skin. This is the hint. Circular sucking mouth, cyclostomata. Now, paired fins and scales, they were absent, right? So it is clearly cyclostomata. So petromyzin is the example, scolidon is chondrichthyes. So this is your exocetus flying fish, and this is branchiostoma, not even a vertebrate. So next part, they are saying select the correct statement regarding the spermatogonia. They always undergo meiotic cell division. Always. Again, question mark. No, beta, they have to increase their cell number. They will also undergo mitosis. So here in this option, A is there, I eliminated that. A is there, I eliminated that. Now, primary spermatocyte, beta, it divide mitotically. No, beta, it is the one that will undergo meiosis 1, and you will get the secondary oocyte. So obviously B is also incorrect. I got my answer, that is C and E. Next part, beta, this student has marked it incorrectly. What is the probability of having children with O blood group? Acha, okay. Where mother and father are heterozygous. Heterozygous for A, B. So AB, heterozygous A, heterozygous B, O. So obviously 25% should be the correct answer here. What should be the correct answer here? 25%. Now, here, right, I'm well prepared here with the answers because we want you to provide the accurate content is here. So you can see it is as per the RAS mechanism. So we know that when it comes to the RAS mechanism, we know it very well what's going to happen. When there is a drop in blood pressure, there will be a drop in GFR, beta. Your JG cells, your juxtaglomerular cells, your granular cells, they releases the renin. And that renin, beta, that renin actually acts on angiotensinogen. What is this angiotensinogen? It is an inactive plasma protein. You will get here what? Angiotensin 2, and then it will be converted to 1 by ACE enzyme, and that will act on aldosterone. So as per that, first will be C. So it is a fall in GFR rate, due to which, beta, what will happen? Renin will be released. So E option, renin. They didn't mention in the options that who's going to release that. So renin will convert angiotensinogen into 1 followed by 2, right? While saying the sequence, I mentioned it incorrectly. Sorry for that. So renin converts angiotensinogen into 1 and then to 2 by ACE, of course, angiotensin converting enzyme by lungs. Then here what's going to happen? Vasoconstriction by angiotensin 2, right? And release of aldosterone. So it acts on the proximal part. We know it very well. And then reabsorption and sodium and water from the distal part. And finally, the blood pressure and GFR will increase. So fourth will be the correct answer here. Now, beta, it is very simple, tidal volume. Even if you know D will go with 2, right? You can, tidal volume. You can eliminate 2 options directly, right? Again, 50-50. So ERV is going to be your what? Expiratory reserve volume is basically your 1000 to 1100. A will go with 3. Then your residual volume, B, will go with 4. Then your inspiratory reserve volume is 2500. So this is the third is the correct option. Now again, IUDs, one question I told you already will come from this. So progestasert, hormone releasing, multiload 375, copper releasing, diaphragm is a barrier method used by females, Saheli is this oral contraceptive. So this is the correct option here. Next part, beta, very simple. It is non-membrane bound cell organelle, which is found in both. It is ribosome. That's why you also call it as universal organelle. That's why you are also going to call it as universal organelle. Now, again, solved in the last lecture, beta. So, ecological pyramid represents the relationship between organisms at different trophic level, and they are generally inverted for. Generally is the word, right? Energy pyramid cannot be inverted. In grassland, number cannot be inverted. Biomass in grassland, definitely not. But in the case of, right, C, it will be. Even I have posted a short video. For the same as well. Next part. Hanji beta. Next part, the flightless bird. They have clearly mentioned flightless bird. But what is the key word here? So it is not like you will read this and you will mark truth here. No, they have mentioned pedal-like structures are there. So this is going to be your penguin. So flippers, right, they are having. So that is why fourth is the correct answer here. Now, beta, again, I told you the most expected question, the most obvious question, and this paper is totally from the previous year questions. So streptokinase, they remove the clots from the blood vessel. Statin is blood cholesterol lowering agent. Lipase, detergent formulation. Cyclosporin, immunosuppressive agent. So 1 is the correct answer. Next part, beta, please look at it. Endomembrane system includes Golgi complex, endoplasmic reticulum, and mitochondria? Not at all. You can even remember it like girl complex, Golgi apparatus, endoplasmic reticulum, lysosome, and we also talk about vacuoles. We also talk about vacuoles. Now, here you can see RER bears ribosome on its surface, true. Both mitochondria and plastid have circular DNA, even 70S ribosome, true. A network of microtubule, microfilament, intermediate filament is there in the cytoplasm, cytoskeletal, true. Then mitochondria is single membrane, beta double. So eliminate E, eliminate A, you got B, C, D. Now, here, set of fishes which belong to class Osteichthyes, that is bony fishes. So your flying fish, let me check again. Sawfish, fighting fish, dogfish is scolidon. We know that Indian scolidon, Indian shark basically it is, right? This is a scolidon. So devilfish, you know that mollusca, this is mollusca. Then hagfish is cyclostomata. So starfish, you know that it is the echinoderm. So clearly cancel that. So third is going to be my correct answer. Now, this question is directly, this question, beta, we have discussed it also. I told you that, right, XX or XO sex determination. So in the case of insects, right, in the case of insects, you people will see females are going to show that, this male will show that. So male will have one less chromosome, female will have more. So beta, whosoever marked it incorrectly. So 23 and 24 respectively is the keyword here. Right, respectively is the keyword here. So lesser number will be there in male, more number will be there in female. So fourth is going to be the correct answer rather than 3 here. Is that clear? Chaliye, next question. Here you can see, I know some of you will think that it is a difficult, actually not, because WBC count they have given. Right, now you know the WBCs, MLA, Benji, that is the trick. You should know the concentration as well, we have discussed that. So eosinophil is around 2 to 3%, lesser is basophil, less than 1%, right? So lymphocytes are 20 to 25%. So on that basis, if you will solve it, so 2 to 3% of 8,000 will be what? 2 to 3% of 8,000 will be what, beta? It is the simplest question, 160 to 240. If you will count this, you will get to know about this as well. So any doubts here? Yes, beta, any doubts here? So if you people are having any right doubts or something, you can ask me, but I hope it is done. It is crystal clear to everybody. Sure, beta? Sure. Done. So 155, this is what we are solving. So I hope it is done. Sure? Chaliye, next part. Now, nicotine, morphine, multiple times we have discussed morphine. I think 3 or 4 times, so I have discussed it, right? So morphine is the pain reliever, analgesic it is. So it is the effective sedative and pain killer. So B will go with 3. I will eliminate 2 option. Now, nicotine, beta, right? It stimulates adrenal gland. It releases catecholamines into blood circulation. But heroin is the another hint. Heroin part is the another hint. For me, it worked, right? Because why? A depressant it is. So second is the correct answer, beta. So cocaine there causes a sense of euphoria, right, and the increased energy. Now tetany, beta, tetany, wild contraction in muscle due to the low calcium in body fluid. So we know that A will go with 3. So you can eliminate 2 options here. Arthritis, inflammation of joint. Myasthenia gravis is autoimmune disorder affecting neuromuscular junction. Muscular dystrophy due to dystrophin gene mutation in that. So progressive degeneration of skeletal muscle. So 1 is going to be the correct answer here. Next part, beta, haploid cells. Right, in which animal do haploid cells divide mitotically? We know that male honeybee, right? So it's kind of repeated. Parthenogenesis it shows, beta, right? Parthenogenesis it shows, right? Here from the haploid gamete it will directly grow into the adult without fertilization. So obviously if it has to form the gamete, it will go mitotically. Next part here. Again, this is the sequence. From the breathing and exchange of gases. For this particular topic, na beta, I just took, I think 2 or 3 seconds. Why? Because cellular respiration should be at the end. At the end, there is cellular respiration. So in all the options, what I checked first, let me cross check now. Acha, two options are there. So I checked the D part. I checked the D part first, D last way. So I cancelled these two options. Now let me cross verify this. Pulmonary ventilation, by which atmospheric air is drawn in, CO2 rich alveolar air is released out. Right, done. Now, after that, diffusion of oxygen and CO2 across alveolar membrane will be there. Then beta, transport of gases by the blood will be there. Then diffusion of oxygen and CO2 between blood and tissue. And finally, the cellular respiration. Next part here. Right, one confusion can be there. They are asking the sequence of layers from outer to inner side. So outermost. So I started with the reverse order. Plasma membrane, zona pellucida, in between perivitelline space, then comes corona radiata. So check the sequence. Corona radiata C, then zona pellucida is A, then perivitelline space is B, and then this is the plasma membrane. So second is going to be the correct answer. Next beta, human protein alpha-1 antitrypsin. Again, I emphasized it multiple times. Right? I emphasized it multiple times. Right beta, emphysema, alpha-1 antitrypsin. Now, select the correct options. Membrane of human RBC, approximately 52% protein. Here I checked mainly one point. They are saying eukaryotic cell. Here they have used the term mesosome, which belongs to prokaryotes. So all three, they were showing the C. I cancelled it out. I got my answer. So A is correct. B is also correct. D is also correct here. So that's how we opted the answer. Next part beta, male frog can be distinguished. Right, I don't know whether you have studied the frog or not. Two questions were there. Beta, male frog, vocal sac, copulatory pad. Right, this is what I located here. Vocal sac, copulatory pad. Right, that's how male frog can be distinguished from the female one. So B and D, vocal sacs are in male, copulatory pad on first digit of forelimb is in the male. Now, this part, if any of you, right, I always discuss it in this way beta, dn over dt, rn, this is the Verhulst-Pearl logistic growth curve model, S-shaped growth curve model. So it is कान के नीचे कान, that's in Hindi. So कान के नीचे कान, K minus N over K, right? So this is the environmental resistance. That's how I discussed. Kaan ke niche kaan. So this is going to be our correct answer. Now let's move to the next part. You can read it as well. Hepatic portal system is a venous system that connects the liver and intestine. Solved it, right? There are 12 pairs of cranial nerves. 12 pair of cranial nerves you will see in the birds also. In fishes or amphibians, you will see 10 pairs. 10 pairs of cranial nerves. I discussed that too. So B is incorrect. I located that. I cancelled this. I cancelled this. I cancelled this. Baki, rest all are correct, beta. So these are the things. D is incorrect rather. So ureters and oviduct, because female, they open separately into cloaca, because females may, you know that urinary opening and this vaginal opening, it is different. Now hindbrain is having cerebellum, medulla oblongata, but optic lobes are not the part of hindbrain, right? They are basically in the midbrain. So sinus venosus will join the right atrium of the heart. So ACE should be correct. Next part beta. Now this one, let's discuss that, right? You should not have any confusion. Erythroblastosis fetalis. Fetus with Rh negative, no beta. Fetus should have Rh positive. So either A, and they are asking incorrect statement. So because they are asking incorrect statement, that should be in our answer. Rh antigen is observed on RBC in majorly in the human beings. That's true. Majorly is the plus population, means the Rh antigen is present. Before blood transfusion, Rh group should also be matched. It is correct. So BC is correct. So they are asking what? BC is correct. So I just eliminated the option. So they are asking incorrect. Incorrect is the keyword here. Incorrect is the keyword here. Now, erythroblastosis can be avoided by administering, right, anti-Rh antibodies to the mother after the delivery of second child. No, here it should be after the delivery of first child. So one is my cortisol, aldosterone, cholecystokinin and progesterone is given. I'll pick up the progesterone here. I'll pick up the progesterone here. So D will go with 1. So I will eliminate these two options. Now, the next part, aldosterone, just now we discussed beta. Stimulate the reabsorption of sodium and water. So B will go with 3. I got my answer beta. Right, let's cross check. A, cortisol produces anti-inflammatory reactions, true that. Aldosterone stimulate reabsorption of water, true that. Right, cholecystokinin obviously beta stimulates secretion of pancreatic enzymes and bile juice, and D will go with 1. Done. Next, The following are the stages of the life cycle of Plasmodium. Arrange the stages in a proper order. The parasite, proper order. Female mosquito injects sporozoite in humans during bite. So I'll start with E. So I have two options with E. I will eliminate that. Now, sporozoite reach the liver through blood, ED. But here C is also given. Let me check. No. First, there will be D itself, beta. Now, let's check this answer. Let's cross-verify that. Then comes B. Parasite reproduce asexually in liver, bursting the cell, release into blood. A is saying parasite reproduce asexually in RBC. First liver, then RBC. True that. Then gametocyte develop in RBC. Done, beta. Second is going to be my correct answer. Next part, we are about to finish the biology. So digestive system, incorrect they are asking. This is my keyword here, incorrect. Pseudocoelom is possessed by Askelminth. This is correct. So C, I'll eliminate. I will eliminate. So B is there in both. So I will not even read it. Right. And I have to go for D or E. Notochord is persistent throughout the life in the class. Chondrichthyes, acha. Beta chondrichthyes, vertebrates. Right. D is my incorrect one. So D is also incorrect. D is incorrect. And they are asking incorrect statement. So bilateral symmetry is the characteristic feature of adult. No. So this is incorrect. Bilateral symmetry, we know that it is in larva. Adult will have radial symmetry. So members of class Reptilia maintain a constant body temperature. No. Even it is also incorrect. Right. Okay. They are asking the incorrect statement. This is beta. Okay. They are asking incorrect statement. Let me choose that again. Digestive system in platyhelminth is incomplete. True. Bilateral symmetry is a characteristic feature of adult. No, adults are having radial. This is incorrect. This is incorrect. Pseudocoelom is possessed by Askelminth. This is true, beta. This is true. Okay, we have to find the incorrect statement. So this one is also true. Notochord is persistent throughout the life in class. No, this is also incorrect. Members of class Reptilia have a constant body temperature. Exception is, no, this is also incorrect. So they are asking about what? Incorrect statement. So let me see B and E. Okay, they are incorrect. C is true. So C will be eliminated. C will be eliminated. So D. So chondrichthyes, beta, and no. So what should be? So let me check that. So notochord is persistent throughout the life in the class chondrichthyes. So chondrichthyes, beta, they are also the, you know that we talk about the subphylum, right? We talk about the let me cross verify that too. Subphylum vertebrata, right? So vertebrata, we know that, you know that we will talk about the Agnatha, Gnathostomata. In Gnathostomata, you will talk about superclass Pisces, and you are going to talk about the superclass what? You are going to talk about the superclass Tetrapoda. So obviously we know that in the superclass Pisces, we have chondrichthyes. So in their case, cartilaginous skeleton is there. Okay, beta. So cartilaginous skeleton is there. So just wait for a while. So this one is incorrect for sure. Otherwise, A or C will be the answer. So they are asking incorrect. I'll cross verify the answer. Let's solve another 3 or 4 questions, then let's proceed. Specific receptors for the neurotransmitter in a synapse are present on, obviously, postsynaptic membrane. Now, a motor neuron. Now, the next question, beta. Again, they are asking the correct answers from the option given below. So, up to this part, all correct? Up to this part, all correct? Yes, everybody. Up to this part, all correct. I believe everything is correct. Yes, everyone, I hope this is correct. Chaliye, let's go. Let's discuss the next part here. So this question is very simple. Try to arrange the sequence as well. And even I will also do it in front of you here only. Just 1 minute. Chaliye, done. Okay, so a motor neuron carries a signal sent by the CNS to the sarcolemma of the muscle fiber. Okay. The neural signal generates an action potential which causes the release of calcium into the sarcoplasm. Okay. Increase in calcium inactivates the actin for braking. So this one, actin binds to myosin head to form a cross bridge, or is it myosin binds to actin, right? Or is it myosin binds to actin? Then beta, then what do you have? Shortening of sarcomere takes by pulling. Basically, you know that it is sliding filament theory, isn't it? Isn't it? So this is the pulling action of actin filaments towards the, is it the pulling action of this actin filament here? Is it so? Done? You tell me. Is it so? So it is the myosin head that will bind with the actin, and then beta it will be, right, because it will be pulled towards the right center part. The H zone will disappear. I hope you know that. Yes, I hope you know that. Yes, everybody, I hope you guys are aware of it. Hanji beta. So any doubts here? So what should be the correct answer here? What should be the correct answer here? You tell me. Which statements are the correct here? Don't you think A is correct? Yes, ma'am. A is correct because the motor neuron will carry the signal to the sarcolemma, and neural signal will generate the release of calcium into the sarcoplasm. And this calcium is required for this initiation. But increase will not break that. This is incorrect. They are asking the correct statement. So I will eliminate these two options, right, beta? Now they are saying that actin binds to the myosin head to form a cross bridge. So you tell me, is it the actin myosin head will bind to the actin, actin myosin, they will form the cross bridge? This is something that we know, right? This is something that we all know. Yes or no, beta? Yes or no? So actin binds the myosin head, cross bridge will be there. So D is also correct. And now when it comes to the E, actin is pulled towards the center, beta, right? It is pulled towards the center here. So here if I have to see the options, right? Here if I have to see the options, actin will bind to the myosin head to form a cross bridge. The myosin will bind to the actin head to form the cross bridge. And shortening of sarcomere will be there by pulling actin filament towards the center of A band. Basically, we are pulling that, H zone will disappear. So I think this A, B, D, E. Yes, beta, A, B, D, E can be the correct answer. But still, we have to see the NCERT statement here. Now, beta, here in this question, right, in this particular question, this part, acha, we have to discuss it yet, or we are done with it? No, not. So this part is done and dusted. So here you can see this one is also incorrect. This one is also incorrect. I told you that, right? And this one is also incorrect. So we have 3 incorrect statements, hai na? We have 3 incorrect statements because members of reptilia is also incorrect. So is it B or D? Because they are asking incorrect. Or is it going to be B or E? So what should be the correct answer here, beta? Chats, please. Chat. Hanji. So beta, here these 3 are incorrect. That we are sure of. So you can see BE and BD both are given. Chats. Done, beta? So here, let me see this because these 3 are incorrect statements that I'm sure of. Chali, this part also. Now, which of the following is not an example of convergent evolution? So we know that analogous organs, they show the convergent evolution. So eyes of octopus and mammals, convergent, this analogous organ, wings of butterfly and birds, analogous, flippers of penguins and dolphin, and four limbs of whale and bat, right? So they are saying which is not an example of convergent evolution. So whale and bat, you know that here what is going to happen, right? Basically their basic structure is same but the functions are different, right? So here second is the absolutely, it is the correct answer. Okay, beta? So now JG apparatus, just now we discussed it. So from renin part they have asked 2 questions, right? From renin part they have asked 2 questions, right? From renin they have asked 2 questions, isn't it? Isn't it? Beta, that is what I'm saying na, D, right? And shortening of sarcomere will be there by pulling that actin filaments towards the center. So that's why I'm telling you this is the answer. Chaliye, juxtaglomerular apparatus is a special sensitive region formed by cellular modification in dash, related to the, if you remember the juxtaglomerular apparatus, you know it very well. Here we have the glomerulus beta, here we have the afferent and the efferent arteriole. So here the distal convoluted tubule will come in the contact. So here you can see DCT, and beta, is it going to be A or is it going to be E? Now this is the keyword. So second and the fourth, obviously they are not there, right? Second and the fourth, definitely it is incorrect. So what is it? Is it going to be A or is it going to be, is it going to be A or is it going to be E? Yes, everybody. Is it going to be A and is it going to be E? Quick. So it's simple. You can even see the option. So if I have to go as per the NCRT, if I have to go as per the NCRT, so obviously I will go for what? I will go for the afferent, right? So I will go for what? I will go for the afferent arterioles, even, sorry, even in the NCRT, my bad beta. So yes, even in the NCRT, we have discussed this part. So it will be with the afferent, the wider one. Efferent is the one that will leave. So it will go distal convoluted tubule and the afferent renal tubules. So now the next question here, beta. So it is the particular activity of the class of enzymes. So what is the hint here? Beta, what is the hint here? The hint here is simple. The hint here is simple, and the hint here is the double bond will be generated. They are simply saying that you are using this particular enzyme and the double bond will be generated. So obviously, right, it is in between carbon-carbon also, XY also. So double bond is the keyword here. So definitely not the isomerase, right? Not even the functional group is transferred. And in the case of ligase also, it is the molecular glue, right? So here, basically, this one, it is going to be, because double bond generation is the keyword here, right? Double bond generation is the keyword here. Now, beta, the next question, mollusca reptilia, okay. Only 2 or 3 questions are left. So wherever we were having the doubt, we will resolve that too. So respiration they are asking. So amoeba, cellular respiration, beta D. Is it with 3? Chaliye, eliminate the option. Now adult amphibians and the mollusca. In the case of mollusca, where will you go? Pulmonary respiration only is the word, then it is B with the 1. Then it is B with the 1. Okay? So B with the 1, we have decoded that. D with the 3, adult amphibians, pulmonary and cutaneous. So mollusca will have the branchial respiration. Remember, Ctenidia. Second is going to be the answer. So reason behind the holes in the Swiss cheese product, it is the CO2 production. Large amount of CO2, but who's going to produce that? Propionibacterium shermani. Propionibacterium shermani. Next question. Next question, beta. Only 2 questions are left. Okay, so now, yes, beta, this is directly, again, this question is from the evolution. This question is again from the evolution, and again, it is the database question. Again, it is the database question. So they are saying about 65 million years ago, 500 million years ago, about 350 million years ago, right? And about 320 million years ago. So you have to match that. Again, it is as such, right? It is from the NCRT. It is from the NCRT. Right, beta? 156, beta, we will cross check, but you answer this first. So A will go with what? So dinosaurs. You remember this certainly the correct answer. Chaliye, next part. Choose the correct statement regarding population interaction. Even in our morning session also we discussed that. Right, bache? So in both parasitism and commensalism, only one species. Parasitism and commensalism. One species is getting benefited, other is harmed. You know that here it is going to be one is benefited, other is neutral. A parasite is benefited, host will be harmed. Host will be harmed here. So you know that here they are asking correct statement, and I'll eliminate these two. Both species benefit in mutualism, true that. Both species get benefited in commensalism. C. Do we have C anywhere? No. In parasitism, only one species benefits, other species is harmed, true that. In commensalism, one is harmed, other is unaffected, true that. So B, D, E is going to be the correct answer here. So we are done with the biology part here, beta. We are done with the biology part here. Now here in this question, beta, B or D. D to definitely incorrect, okay? D to definitely is incorrect. Even E, members of class Reptilia, they are poikilotherms. So here, beta, I'm not going to mark the answer because these 3 statements are incorrect. Chaliye, one or any other doubt we had in any other question? So let me check that. If there will be anything like that, we'll resolve it right away. So this part is done. This part is also done. This part is also done. Right? So this part I told you 3 statements are incorrect. Okay. And sarcomere part also. So yes, any doubts here? Any doubts here? Any doubt here? Quick, everybody. Quick, everybody. So as per NCERT also we will check. But you tell me if there is any doubt. Quick, let me see. Yes, beta. So quickly tell me if there is any doubt. Hanji, so it is 173. Okay, 1 is going to be the correct answer. We have already discussed. So now in this particular part, this one beta surely I will check. So because both are correct now, because 3 statements are incorrect. 3 statements are incorrect. So here majorly we will go for B or E. B or E. But notochord is persistent throughout the life in the class. See, in the chondrichthyes, we know that, right, the cartilaginous endoskeleton is there. So out of all the options here, we can go for B and E, right? We can go for B and E here. But overall, beta, overall, you know that in the case of notochord, in the case of this chondrichthyes, right? What will you see? Notochord is persistent. Is it so? देखो बेटा, why I am going for B and E here? मतलब I'll give you one simple explanation. बाकी let the NCERT come. So no doubt this is correct, right? That chondrichthyes, because chondrichthyes are having cartilaginous endoskeleton. We know that. So this notochord will be replaced by that, right? Like it will be replaced by that cartilaginous endoskeleton wala part as well, cartilaginous vertebral column as well. So notochord is there in the embryo, in the adult, but cartilaginous vertebral column will also be there, right? But remnants will be present, so that's why we are considering this. Chali, so now the chemistry part is going to start. 179. Beta, wait. I'll definitely check. Hanji, so what's the doubt here? What's the doubt here? You please tell me. We'll clarify that. What's the doubt here? Beta, it will go with, okay, A will go with, let me recheck it. Don't worry, let me recheck it. You don't worry about 65 is second, right? B will go with 4, about 500 million years ago. That is also true. So about 350, okay, 350 million years ago will be this, ma'am, and D then will go with our 3. Now fine. Any other doubt? Quick, everybody. So 179, it will go with 1. Okay? It will go with 1. Chali, done? So should I start the chemistry part? Okay, let's start. Let me connect with Gaurav sir. So, Hanji, here, what's your doubt? Yes, beta. That part was important. So 156 में, okay, cry one, oh, done beta, done. So Hanji, which one is the correct answer here? So you tell me, any other doubt? Any other doubt? Quickly tell me. So it is 156. So we will go with third beta, cry 1 AC and cry 1 AB, isn't it? Cry 1, you can communicate here, right? Otherwise it is absolutely right. And if there will be any update, we'll update you, we'll mention that too. Okay. So here let me recheck this once again. So you are going for this corn borer. Corn borer will be this third one, beta. So it will be cry 1 AB and it is going to be cry 1 AC. Is that clear? So this is basically for 156. Okay? So any other doubt? Chaliye, now it's time for chemistry. So let's get started, beta. So, okay, so chemistry time is here. Done? Chemistry time is here. So this statement, beta, right, here in this statement I have clearly specified, right? So do remember the depth here of this particular question also. Baki NTA will provide the correct answer as this only. So you can see that. So this part is also done and dusted. Okay, everything is clear. Done? Everything is clear. Any doubts? Chaliye, so Subrat, okay, done.
Now let me finish with chemistry, because yes beta, now I would rather say I have analyzed the complete paper and it is quite doable, quite doable. Yes, you just have to be very much vigilant. Just a second guys, just a second. Hello. Hello. So, guys, let's start. Take out your pen and copy, take out your rough copy, and let us start the process of attempting the whatever chemistry we have. Let us finish the complete chemistry. Now, the few questions I have discussed, but yes, the arrangement of paper definitely takes time. So this is our next question, a very simple question from chemical kinetics, and once you have your paper arranged, so obviously it becomes very doable. So let us start the process. The question is, you have to relate rate of reaction with unit. So, sir, definitely we know rate of a reaction is equal to rate constant into concentration raised to the power order. So, what can you say about rate? Rate is change in concentration upon change in time, and K is definitely, sir, that means that will come over here. We remember this. You do not have to do this in examination. Then, sir, why you are telling us like this? Because I want you to remember so that whatever students are here, हाँ बेटा, chemistry was, I would not say wire fire, will fire. It was not hard, but yes, you have to be very much very much comfortable with your content. And if at all you are comfortable, then only you are going to make your answer correct in your examination. है ना? So it was doable. It was doable, but yes, lengthy. I mean, you if you have practiced enough, then only you will be able to attempt your paper in a limited amount of time. So, sir, for a, let's say if n value is 0, that means this is gone. So, rate constant will be concentration upon time. So, for a zero order reaction, what is unit of concentration? Mole per liter, and for time, let's say per second. So, that means A is definitely 4. So at least one option is eliminated. Now for a first order reaction, this is gone. So obviously we remember this is only for the time. So this is just per second. So that means B is definitely 3. So that means this is also eliminated. So I'm left with 1 and 3. And obviously for a second order reaction, if you do, so second is -1. So everything just get reverses. So I guess for a second order, it will be mole inverse liter second inverse. That means 1. So I guess you will be landing up with, yes, C goes with 1. That means 1 is incorrect. Okay. So I hope we have got our answer. It is C. Done, guys? Chalo. Chalo. Moving to answer for the previous question was definitely C. I hope everybody got the answer. Guys. Chalo. Moving to the next one. A bulb at 140, okay, the power is given and its efficiency is 8%. Fair enough, sir. Whatever it is having power, what is the effective energy? What is the effective total energy, sir? Power is energy per unit time. So let us take it for 1 second. So what will be the total energy, sir? The complete is not giving, the source is not giving it to us. It is giving us just 8%. So 8% will be definitely 1.5 into 8. So this much of joule of energy we'll be having. So this is definitely 15 into 8. This is the calculation is, this is 120. So this is 12 joule. Now, if energy of one photon is given out to be this much, then sir, this is the complete energy, and complete energy is equal to number of photons into energy of each packet. So what is the energy of each packet that has been supplied to you, sir? It is 4.42 into 10 raised to the power minus 19. And can you calculate the number of photons? So now you just have to do the mathematical calculations, and I guess this will be easy also to calculate, and once you'll solve, you'll get 3 as your right answer. I hope everybody is with me on this. Guys, chalo, perfect. Moving to the next one. Yeah, the next question from physical chemistry says the number of hydrogen atoms present in 5.4 gram of urea. Now what is urea? Sir, urea is NH2 with a carbonyl group, then a NH2. So this one molecule of urea, is having how many hydrogens, sir? 4 hydrogens. So I just have to calculate the number of urea molecules. So number of molecules upon Na will be equal to weight of urea, which is 5.4 gram upon molecular mass. Sir, you can obviously calculate. We already remember it. It is 60. So if it is 60, I can calculate number of molecules of urea. So number of molecules of urea will be 5.4 upon 60 into 6 into 10 is power. I'm approximating it. So this will be number of urea molecule. But are you asked number of urea molecule? No, sir. We are asked number of hydrogen. So number of hydrogen atoms will be 4 times number of urea molecule. I hope everybody is with me on this, guys. Guys, I hope everybody is with me on this. Now, obviously, if you just solve this, you will get somewhere around 1. Yeah, into divide I'm not doing for you, because yes, this is the job that we have done back end and we have validated, because I know most of you must be eagerly waiting, sir, answer. What is the answer? So obviously I'm giving the answer also, but I'm giving the logic also for all the other students who will be watching this in the recorded format, so that it will be give them the conceptual understanding as well. I hope everybody is with me on this. Perfect, guys. Chalo. So for the students who are saying fast, I'm giving answer also, and for the students who will be requiring this for their knowledge, I'm giving you the concept also. Chalo. Moving to the next one. Phenolphthalein is used as an indicator for titration. Sir, we definitely know if we have phenolphthalein, then in acidic medium. Now the question is asking you titration of sodium hydroxide solution against a standard solution of oxalic acid. So what standard solution I have? Sir, I have oxalic acid. So that means it will be acidic medium. And once you use phenolphthalein, medium will turn basic. So in acidic medium, color of phenolphthalein is colorless. And once it acquires basic medium, the pH range is somewhere around from 8 to 10. But yes, at the neutralization point, even with a drop of the reagent, the pH changes drastically. So from 8 to 10, it changes drastically, and color changes from colorless to pink. So your answer will definitely be option 3. Yes, this is a student whose paper we have taken. But yes, this will definitely be from colorless to pink. I hope everybody is clear with this, guys. Chalo. Which brings us to the basic question from thermodynamics. Sir, a question from float, first law of thermodynamics. So what is float, sir? First law of thermodynamics. What does this say? At a certain temperature during process, 500 joule is absorbed. So that means Q is plus 500. Work of 200 is done by the system. So work done by the system is taken with a negative notation. So this is minus 200. Can you tell me what is change in internal energy? Yes, sir, this is easy, Q plus W, and we know the value. So this is 500. So this is just 300. So option 2. Option 2 is your right answer. I hope everybody is good. Good. Guys, chalo, perfect. Moving to the next one. Sir, again, Faraday law of electrolysis. So what does this Faraday law of electrolysis is gonna tell you? Sir, W is equal to weight of any substance deposited is equal to ZIT. So it is equal to electrochemical equivalent into charge into T, and we just have to calculate the weight. What is the element we are interested in? Sir, we are interested in the electrolysis of copper. So how can you calculate the copper which will be liberated? Sir, copper will be equal to Z, which is E by F into I into T. Sir, what is E? It is the equivalent weight of the substance. What is the substance we are talking of copper? So molecular mass of copper, I mean atomic mass of copper upon its valency factor. So what will be the value of E? Let me write E separately for you. It is atomic mass upon valency factor. So E for copper will be 63 by 2. Faraday number is charge on 1 mole. So it is 96500. I guess current is given out to be 1.5 amperes. And time in seconds. This is 10 minutes, but SI unit of time is seconds. So 10 into 60. Yes, you have to do the multiplication by yourself, and once you will solve. Yes, I will not say this was an easy question. I will never say this was an easy question, because it requires calculation. You have to put in values also, and it requires calculations. So you should not attempt such questions in your first attempt. Always have an habit of attempting these questions on the second go. I hope I'm making sense over here. Chalo beta. Moving to the next question. The next question says, you beta, such questions are very crucial. Now why I'm saying crucial? Because see, it is asking you correct. Sometimes we misjudge that whether it is asking correct or incorrect. So it is just asking you that how many of the options are correct. So let us attempt those correct options. How many of them are correct? The molarity of 2.5 gram ethanoic acid with molar mass 60 gram in 75 gram benzene is, sir, that means the value is already given. The value of what is asked? Sir, molality, as in small m. So let us just write the formula of molality. So I'm doing this for part A. Molality is equal to weight of solute upon molecular mass of solute into weight of solvent. Now solute I write as 2 and solvent as 1 because I'm putting this weight of solvent in grams, so I add 1000 in the numerator. So molality, we have to check whether it is 0.55. What is weight of solute? Sir, it is 2.5 grams. What is molecular mass of ethanoic acid? It is already given 60. How much solvent I have used? Sir, 75 gram. So it will be 1000. And definitely once you will solve this, I'm sorry, it is 1000. And definitely once you will solve this, it is correct. So correct, that means A is definitely your answer. Now after that, molarity of solution of 5 gram of, or let's say just go to the options. If you check B is there in second option and fourth option. D is there in only this option. And again, so just check for C and D. Just check for C and D. That will also give you an idea. That will also give you an idea. So let us go to the options directly. This is a theoretical option. You do not even have to do any calculation. For binary mixture A and B, the number of moles of A and B are Na and Nb. It is defining. Now it is saying mole fraction of B. Sir, mole fraction of B will be Nb upon Na plus Nb, but it has written Na. That means this is incorrect. So because it is incorrect, so third cannot be your answer. Now let us eliminate one more option. So let us check for E is not there. C. Okay. Let us check for C. Aquatic species are more comfortable in cold water. Sir, if you have water cold, that means pressure of gas, that is Henry's law. So if you just apply Henry's law, as you increase the partial pressure of oxygen, it will be more dissolved in the water as well. And if you increase the temperature, the gas will flow out, because as you increase temperature, gases they attain okay, as you increase temperature, they attain kinetic energy and they flow out. And if temperature is less, then they will remain in the solvent. So answer for this question will definitely, sir, C seems correct, because aquatic species will feel more comfortable because at lower temperature, kinetic energy of gas will also be low, and therefore oxygen gas will be there in the solution. That's why they'll feel comfortable, because more oxygen is going inside their system. So definitely C is correct. So because C is correct, so this can be your answer and this can be your answer. But yes, we have to check then only for E, for option Delhi. The solubility of gas increases with, no sir, for temperature we can say, but if you decrease, the solubility of gas increases with decrease in pressure. Sir, if you increase pressure, then their solubility will increase. Why? Because this is Henry's law. Partial pressure of gas is directly proportional to mole fraction. That means their solubility. So obviously D is definitely incorrect. So option 2, let me use another pen for this. So option 2 will be your right answer, because A is correct, B we have not checked, C is correct, A is correct, B we have not checked, but I guess we are not left with any option. Obviously, if you check also the formula of molarity, the only reason I'm saying this, because this was a time-taking process. This was definitely a time-taking process, and the reason I'm doing this is because I don't want you to again do, sir, weight of solute, molecular mass of solute, weight of solvent, volume of solution, because volume of solution is also given and it is calculative also. So that is why I was searching out a way so that you should not again solve this and eliminate option, and that option elimination can obviously help you. I hope I'm making sense over here. Yes, it could have taken time. So your hustle for saving time is rather an option that you should always pick as a way out to save your time and increase your efficiency during a D-Day. Chalo. Okay, moving to the next one, beta. Mixture of chloroform and acetone. Now, this is straight a theoretical NCERT questions. Mixture of chloroform and acetone. Sir, we definitely know that this is chloroform which has very strong hydrogen bonding. So the very strong plus and minus, sir, but carbon is not electronegative. I hope this is an NCERT logic and everybody knows this, that this will have strong minus I effect, and that strong minus I effect will make this carbon also electronegative. So there will lie or exist a very strong intermolecular force of attraction between this minus and this plus. So this is your hydrogen bonding. So formation of hydrogen bondings, obviously option 1 is only giving you the right answer. So I hope everybody got it. A very simple and doable question, not taking much of time as well. Okay. Now, after this, now at 298 Kelvin, a buffer solution contains equal concentration of X minus and HX. So that means it's an acidic buffer. Acidic buffer is weak acid and it's salt with a strong base. So what is pH of an acidic buffer, beta? pH is equal to pKa plus log of salt as in the conjugate part upon acid as in HX. Because both their concentrations are given to be same as an equal, so they are gone. So log 1 is definitely 0. Now we just have to calculate Ka. So Kb is given. So for conjugate acid base pair, so Ka will be equal to Kw upon Kb. At 25 degrees Celsius or 298 Kelvin, sir, this is Kw stands for minus 14. So minus 14 upon minus 10, that means this will be minus 4. I hope everybody is clear. Now, so pH will be minus of log of 10 raised to power minus 4. So your pH will be definitely 4. I hope everybody is getting this. So your answer is definitely option 3. Is it good, guys? Good. Chalo, moving to the next one. Moving to the next one. Again a simple question from chemical kinetics. I told you initially only that you will be getting question from Arrhenius. We got question from Arrhenius. You will be getting question from zero order, first order. Sir, zero order. What is this? Sir, concentration is decreasing with a slope. Sir, we know integral form of rate law for zero order reaction. Sir, it is AT is equal to minus KT plus A naught. So it is a straight line with slope value of A naught, with slope value of A naught, with intercept as A naught, that is initial concentration, and slope of minus K. So this is definitely, the student has also done it correctly. This is definitely a zero order reaction, a very easy questions. Good. Chalo. Okay. Good. Next. I hope everybody is done with this. Moving to the next one. Now, again, a basic question from thermodynamics, but yes, this is also calculative, so I would not advise to do it on the first go. Do on the second half of your paper. First attempt the easy questions, because I'm not saying it is difficult, but yes, calculations. The reaction is given, and for this reaction, you can definitely calculate delta G. So delta G will be equal to, of product is 2 minus reactant is 2 plus 1 as in 3. So delta G will be minus 1. What is the relationship between delta H? Actually, you have to calculate delta G. So delta G will be equal to delta H minus T delta S. Temperature is also mentioned, 298 Kelvin. Delta S is also mentioned, minus 44 joule. But sir, delta H is not given, and instead of delta H, delta U is given. So here you have to calculate delta H. So delta H will be equal to delta U, which is minus 10. But it is in kilojoule, mind you the unit, so it is calculative. Delta H is equal to delta U plus delta Ng, which is minus 1, so minus R into T. R is definitely, in terms of joule, it will be 8.314, but you have to do a division by 1000. You know why? Because this unit I have taken in kilojoule. So you have to convert that into kilojoule. So actually, you have to calculate delta H over here. It will be coming in kilojoule. It will be coming in kilojoule because I'm dividing 1000. What will be value of R? 8.314. What is T? 298. So yes, it is calculative. You can approx also, but still after approxing also, you have to do that calculation. So you will be getting a value of delta H in kilojoule. So here I am getting delta H in kilojoule, but again over there also temperature is 298 and delta S is given out to be minus 44, but value is again in joule. So you again have to do a 1000 division over here. And now you will be getting your value of delta G, and that will also be coming out in kilojoule only, and option is also asking in kilojoule. But yes, once you do, delta G will come out to be positive. And what positive value? 0.6 something. So you can approximate because options are far enough, but yes, calculation is required. So that calculation part I'm leaving. You can obviously do that. Not easy, but yes, in a paper, not at all easy. Why? Because time is a constraint. So yes, but in back at home, and once you are done with your easy level of questions, and coming back to such questions and attempting them is always advisable. Okay, guys. Hello. Chalo. Moving to the next one. Now, this was a very doable question. Should be killed on the first go. The question says you are given with reactions and you have to relate Kp. We definitely know Kp is equal to Kc into RT raised to the power delta Ng. What is delta Ng? Difference in moles of product and reactant. So what number of, if I want them to be not equal, that means I want delta Ng not to be equal to 0. Because if that is 0, then Kp and Kc will become equal. So let us check for the options. Here this is 2 minus 1 and 1. So this is delta Ng over here will be 0. So not an answer. Here also 2 minus 2. So this is also not an answer. Obviously this is 2 minus 4. So here it will be minus some negative value. So obviously it is not equal. You are just supposed to tell that it should not be equal. So obviously it will not be equal. And over here also it will be equal. So not an answer. A very doable question, a very easy question. Good. Chalo, moving to the next one. Given below is an expression for rate constant. Okay, sir. Again the same thing. What equation? I have even done this in the morning. The same question I used, the same logic I used. What is the relation? Sir, K is equal to Arrhenius constant minus activation energy times of RT. Everybody knows this. So ln of K will be equal to ln of A minus EA upon RT. Sir, just compare the value. Okay, beta, comparing the value. Instead of E upon R, leave this minus because there is also a minus and I need a minus over there. So just leave the minus. So instead of EA upon R, I'm not writing T because T and T both are here. What I do have, sir, I have 1.25 into 10 raised to the power 4. What do I need? I need energy of activation, but in kilocalorie. Just look at the unit of R, sir. R is approximately 2. So take it approximately 2. So EA is equal to twice of that value. So 1.25 is approximately 2 point something or 2.5 something into, but sir, this is calorie. I need kilocalorie. So 10 raised power 3 I'm snatching and I'm saying it is kilocalorie. So 2.5 into 10 is approximately 25. So do I have any option close to 25? Yes, obviously option 4. So this is not calculative also. That's why I'm doing calculation in this question, because this is not calculative also, because here your common sense is going to help you. So that's why I've done those calculation where you have to do those religious class 9-10 calculations. Only that I'm leaving. If in a question the calculation is also with a common sense, I am explaining that also for all those students who will be listening to this as a recorded format. And obviously for the students who have just completed their NEET examination, that's why I'm being quick too, so that you can at least check your answers, because obviously that will be a cause of anxiety inside you. So just get this over with and you can take your day off, the remainder of the day off. so do us. Chalo. Now, okay. Okay, chalo. Moving, acha, physical finished. Now, physical was the only area which could have taken more of your time. I would rather say organic and inorganic was quite doable. So let us quickly finish, complete organic and inorganic, and then we'll be moving to the other subjects. Now, two products. Now, few questions of organic I have discussed once. I've taken the different format of this presentation, and now I have all of chemistry questions with me, divided as physical, organic and inorganic. So physical we have done. Now we have started with organic. Few of the organic questions we have done in the initial phases, so you can obviously watch that in the recorded format. But now the remainder of the organic questions are here. Two products X and Y are formed. So these are X and Y. Okay, sir. Benzene with methyl chloride, anhydrous AlCl. Sir, this is Friedel-Crafts alkylation. So W will be what? Sir, W will be toluene. Easy, sir. Dilute HNO3 and H2SO4. Sir, this is nitration. So nitration will bring what, sir? Nitration will bring NO2 plus, and NO2 will be substituted at para also. Yes, ortho will be less, but still, because this is an activating group, so NO2 will be introduced at ortho position also. So these are ortho and para derivative, which are isomers, we know. And obviously we know these ortho and para isomers have, so the masses are same, they are isomers. So how can they be distinguished? Sir, in order to distinguish, you do distillation. Generally, organic compound are steam distilled. But if you just see the option, then because they are not giving you steam distillation, so rather I am picking the most, or else the best possible option should be steam distillation. But yes, because it is not saying, giving you an option of, because it is not giving us an option of steam distillation, so that's why I'm using fractional distillation. I hope I'm clear with this, guys. Guys. Chalo. Moving to the next one. Now, next question says select the reagent that reduces nitriles. Now, what is a nitrile? Nitrile is RC triple bond N. I have to reduce it to form a 1-degree amine. So, what shall be my list of reducing agents? It's easy. LiLH4 strong reducing agent can do this. Why hydrolysis? Because LiH4 first give H minus and then give H plus. So H minus goes to what? So this is minus, this is plus. So H minus goes here, and after hydrolysis, H plus goes here. Everybody knows this now. So this can be, sir, metal plus acid, sir, it reduces nitro group. Metal plus acid reduces nitro group, so it is not nitrile, it does not reduce nitriles. Yes, it reduces nitro group. So yes, so B can't be my answer. Okay, then sir, catalytic hydrogenation can obviously reduce everybody. Catalytic hydrogenation is a very strong reducing agent. Sir, zinc sodium amalgam in ethanol, this is Mendius reagent, and this can also do that. So sir, this is also my answer. So A, C, D. What about B? Sir, this is classic Hoffman bromamide degradation. So that is a CO is removed from an amide, so obviously this is also not an answer. So now we obviously have seen it is ACD. I hope everybody is clear with this, guys. Chalo, moving to the next one. During Lessaigne's test, sir, what is Lessaigne's test? I have an organic compound, and with that organic compound, I make a sodium extract. Sir, sodium will force all the carbon, nitrogen in form of their, so they are precipitated or extracted in form of their Lessaigne's, the Lessaigne's extract. Now this Lessaigne's extract is nothing but it is a sodium fusion extract. It is a sodium fusion extract. And that extract is definitely, my friend, an ionic compound. That's why I further do the qualitative analysis and can detect the presence of those carbon, hydrogen, and whatever element it has. So whatever extract we have, they convert this organic compound, which is a covalent compound, into it ionic form. So this student has also attempted it correctly, and yes, this is the right answer which the student has also given, and you should also give once such questions come to you. The pair of molecules that are metamers. Now metamers, sir, you have a divalent functional group. Nature of alkyl group should change. Sir, alcohol is not a divalent functional group. Yes, ether is. So this is methyl and propyl on one side. Sir, this is ethyl and ethyl. A very classic example of metamer. So yes, this is a metamer. Moving to the next one. Now, again, a very doable question. Again, a very doable question, because you just have to calculate the sigma and pi bonds. So just do it for, do we have an option which if we do, sir, 1, 2, 3, 4. So just do it for D. Why? Because you do not, if that, then you do not have to check for any, because D is different for every option. So just do it for D. What is D? D is ammonia. So ammonia is, sir, 3 sigma bond and 1 lone pair. Do I have that anywhere? Yes. So that means D is 2. That means, that's why you should always have an open eye. See, it is helping. You have saved a lot of your time. Moving to the next one. Remember in the physical questions how much time we were taking? That's why I'm saying you should leave such questions in the first go. How much time we took? Nothing. Now, after this, compound P. Sir, this is C8H8. Obviously, sir, it should be satisfied by DU. So it will be having one benzene, so 6 carbon gone, but sir, it will have 2 more carbons, and it is saying it gives red orange PPT with 2,4-DNP. That means it should have a carbonyl group, and it does not reduce Fehling reagent. That means this carbonyl group should not be an aldehyde. So make it a ketone. So in order to make a ketone, I need a double bond, but I need carbon on this side also. Then 8 carbons are employed. Obviously it is have 5 hydrogens, so 3 hydrogens. So this is definitely acetone. So that means P is acetone. So not this, not this. Now, on drastic oxidation with chromic oxide, P gives an, okay, that means vigorous oxidation. Sir, vigorous oxidation will break it as per Popoff's rule. So that will be removed in form of CO2 and H2O, and this will be benzoic acid. So benzoic acid. So option 2. Option 2 is your right answer. I hope it is good. Easy one. Now, moving to the next one. Yes, good. Moving to the next one. The functional group that can be, the functional group that you have to identify through, yeah, sir, dye. It is there in NCERT. Dye test is given by phenols and aromatic amines. So phenols and aromatic amines. I do not find aromatic amines, therefore phenol. Although you do not know nothing about this, the Thylene dye test, although this is taught in class 12th in your alcohol chapter also, it is also there in your NCERT lab manual. But even if you do not know this, then from your chapter of amines, you know that the diazonium coupling reaction from the chapter of amines, who undergo and gives those dyes, there may be some other color dyes as well. So phenol and aniline. So this is definitely a test for phenolic group. I hope everybody have got the answer. Easy one. Now, again, a doable question from biomolecules. Sir, what is the, I teach my students a rhyme. DNA has 4 bases. I remember ECG. Because I mean we take our elders for their ECG. So do not say ECG, say ACG, because DNA contains TACG as the basis and RNA contains UACG as basis. So I remember it like this. So ACG is the same. Thymine and uracil, they are the difference between DNA and RNA. And obviously if you just go browse the options, so thymine it is saying, but DNA is the version which is having a double stranded ring structure where the 2 strands are attracted to each other by hydrogen bonding. So DNA possesses a double strand. It is saying it possesses a single strand. That is wrong. Therefore option 3 becomes your right answer. Yes, for these are difficult questions for my JE students, but yes, for your bios, I mean for you, obviously you will be very much comfortable, and I hope you have attempted this one clearly. Moving to the next one, sir, again a very attemptable question. We just have to number methyl is at 3, ethyl is also at 3, but as per rule of alphabetization, ethyl wins. So this will be 1, 2, 3, then 4, then 5, then 6 and 7. That means 3 I have methyl, 5 I have methyl, and then heptane. That means option 3 is your clearly right answer. Moving to the next one. Again, a very doable question, beta. Ethyl chloride reacting with X producing Z. Now Z is a foul smelling compound. Everybody should remember carbylamine reaction. So the SZ must be a carbylamine, which is a foul smelling compound. The second reaction is confirming the same. This is amide. Sir, Hoffman bromamide degradation will remove this amide, and Y will be C2H5NH2 as in a 1 degree amine. Now, when this 1 degree amine once undergo reaction with chloroform, sir, this is classic carbylamine test. So C2H5, and through the chloroform, I'll be getting an attachment of the isocyanide. So C2H5NC will be your Z. So we have already identified this Z, which is C2H5NC. That means these two may be your answer, but yes, I have to form this using a alkyl halide. That means what nucleophile I need? Sir, I need lone pair on nitrogen. If I use an ionic salt, it will be carbon having extra charge, so I cannot use this. I need a covalent solid. I need a covalent solute, because AgCN will not ionize completely. So who will have the extra lone pair to donate? Nitrogen, and therefore ligation, this ambident nucleophile will attach through nitrogen, and isocyanide is produced. So I hope everybody is clear. Fourth is your right answer. Moving to the next one, guys, quickly. Organic also we have finished. Quickly moving quickly to inorganic side of chemistry, beta. Now, this was a question I have just left. Yes, because at NCERT they have removed block chemistry, but still they are asking these things. So that's why I have again taken this question. Yes, V2O5 is definitely, sir, it was a block chemistry logic, beta. In contact process, it was given in NCERT that V2O5 is a catalyst which first oxidizes SO2 to SO3, and post absorption of water, oleum and H2SO4 is formed. So yes, V2O5 definitely goes with 3. Now, iron, if you talk of iron, then iron is definitely a catalyst for Haber's process. Yes, it uses molybdenum and other substances as promoters at specific temperature and pressure, but yes, iron is used as a catalyst. I hope most of us know this. Now, PDCl2, this is also, I mean, not a very famous fact, but yes, from the chapter of class 11th inorganic chemistry, you can see that it is used as a oxidizing agent which oxidizes alkyne to corresponding aldehydes and ketones. Technically, it is used to produce aldehyde only. So from ethyne, it produces ethanol. So yes, again, not a very common usage, but yes. So that definitely C goes with 4. Definitely C goes with 4. So option A, 1 and 2 are wrong. Now, what about D? Nickel complex. Nickel complex is classic used for preparation of alkynes. Again, not a very famous catalyst which NTA has asked a lot of times, but yes, that's why I'm saying this question was, I mean, for a basic NCERT reader, it would be a hard nut to crack. So your answer will be obviously option 4. I hope everybody is clear with this. Now, such questions I would not say are good, but yes, if you have completely read your NCERT, so that ways I would not say that is why I left this question once I was dealing it initially. Yeah, na? Chalo. Now, moving to this question, again a very doable, a very conceptual question. We have to calculate spin. So spin only magnetic moment require number of unpaired electrons. Sir, titanium is 4s2 3d2. So titanium plus will lose the 4s electron, so it will have 3d2. It is showing you also 3d2. That means how many unpaired electrons it has 2. What is magnetic moment? Under root n into n plus 2 Bohr magneton. So 2 plus 2 is 4, 4 into 2 is 8. So that means under root of 8 Bohr magneton. So any answer slightly less than 3. Any answer slightly less than 3. Any answer slightly less than 3. The student is also right. You should also be right. A very doable question. 1 is your answer. Now, in a test tube containing salt, a few drops of dilute sulfuric acid was added, which gave colorless vapor having smell of vinegar. I hope it is done. Vinegar. Vinegar. The very, from class 10th we know this is nothing but a test of acetate ions. A very easy question. No, to do crime to not attempt such questions in your examination. Moving to the next one. Identify the incorrect statement. Carbon has p-pi tendency. Yes, carbon has a p-pi tendency. Why? Sir, alkene, they are formed because of the p-pi tendency. So it is asking incorrect. Because it is correct, so not an answer. ECl3, when forms a monomer, okay, it is saying that BCl3 exists in nature as monomer, but once you have aluminium, so once you have aluminium, so AlCl3 exists in nature as dimer. Yes, this exists as dimer because chlorine having lone pair donates their electrons in the empty orbital of aluminium, because aluminium has vacant d orbital. But this boron guy, this do not have that. Yes, sir, but still this is Lewis acidic. It will also want, at least it has the vacant 2p orbital. So that 2p orbital can be satisfied with the back bond. That 2p empty orbital, that Lewis acidity, can be satisfied by back bond, and this back bond will be effective also because this has small, so because small atomic orbitals have higher tendency to do p-pi back bonding, so the back bonding will be most efficient, and hence it exists as more. So this is also correct. So sir, this is correct, so not an answer. Again, see, a block chemistry logic. Now, oxygen, sir, in chemistry don't use only R. Only -2. Oxygen in with fluorine it show +2 also. In peroxide it show -1 also. In superoxide it show -1/2 also. So yes, in chemistry we don't say only. So because it is saying only, therefore obviously incorrect. I hope everybody is clear with this. Moving to the next one. Now, although +3 oxidation state is not common in lanthanoids, cerium, acha, a very famous question. What is, sir, cerium is the first element. So how do you fill, sir? First we fill 6th period as in 6s2, then we fill lanthanum, that is 5d1, and then comes cerium, which is 4f1. So this is the ground state electronic configuration. Sir, cerium +4. Technically, we know all the lanthanoids primarily exist in +3 oxidation state. But yes, few of them also exist, although they also want to rush back to the +3 oxidation state. But why they do exist? Because they exist whenever they attain a very strong electronic configuration. But still, after attaining that very strong electronic configuration, they still have a tendency to come back to the group oxidation state, which is +3. But yes, in that sense, they can at least exist with very less amount of existence, but they can at least exist in +2 or +4 oxidation state, all the lanthanoids, only when they acquire some special electronic stability. And obviously you can see, once cerium loses all these 4 electrons, its electronic configuration becomes inert. Hence it exists, but still it is not the stable version. Stable version is again the group oxidation state. So the Se, not Cr, Ce. So Ce +4 also have a tendency to rush back to +3 state. That means it wants to reduce itself. So it will behave as a very strong oxidizing agent. Also, these things were not asked, but still very important logic. So what shall be? That means obviously the student has also marked. I am just giving you every detail regarding it. That much was not required, but yes. After losing one electron, it acquires 4Z0. That's why it exists in +4 oxidation state. I hope everybody remember this. Now, moving to the next one. Again, an incorrect statement you have to tell. So I guess you just browse through the options, because whenever incorrect, some statement is highly incorrect. So nitrogen can form p pi d pi. Arey yaar, nitrogen is a second period element. It has no d. How can it form p pi d pi? That too with oxygen. Nitrogen can have a pi bond with nitrogen as p, but other element should have d. But other is also oxygen. So with nitrogen and oxygen, both do not have d. So therefore, Obviously incorrect. I hope everybody is good with this. So to identify such incorrect questions, beta, try to scan the options rather investing completely in reading the complete options. Scan the options first, because sometimes reading those options will give you easily the odd one out entity, है ना? Now, methane reacts with steam at 100 degrees Celsius, a classic reaction, but again, but in hydrocarbons it is given in block chemistry also. So leave block chemistry, but hydrocarbon is there in your syllabus, so you should definitely know this. So methane react with steam as an H2O gaseous at 1000 degrees Celsius will lead to the formation of water gas, will lead to the formation of mixture of carbon monoxide and hydrogen. It is very famous name for that is water gas. I hope everybody is clear with this. Now again, an incorrect statement. So incorrect, again scan the options. Sir, IUPAC, sir, this is un as in 1, nil as in 0, sept as in 7. So this is definitely, because this is correct, so not an answer. Largest and smallest. Okay, these are, sir, largest and smallest. More be the cation, more the smaller it will be for isoelectronic species. So because I have to deal with Al plus 3 and Mg plus 2, because they are isostructural, this has stronger protons, so more be the cation, it will be smallest. So smallest will be actually aluminium plus 3, because cations are generally smaller. Now, who will be larger? Obviously the parent. Who are the parents we are talking of? Sir, we have to compare magnesium and aluminium. So among magnesium and aluminium, sir, it is lying along the period, and along the period we know size decreases. So who should have highest? It should have largest size. So that means the largest should be magnesium. Acha, it is saying aluminium, so obviously this is wrong. And the smallest will be aluminium. So this is also wrong, because smallest will be aluminium. So both of them are wrong, and therefore this is an answer. Why bother? I mean, every option will definitely take some time of yours. So why bother? Because we have obviously attempted the question which was the option which was incorrect. I hope it is clear. Moving to the next one, guys. Now, the correct order of increasing metallic character. Sir, again you just have to draw that periodic table. So let us draw that periodic table. Sir, so beryllium, second period, second group. After that, here I have sodium, because lithium, beryllium. So after that, in the next period I'll have sodium, and after that in the same period I'll be having first phosphorus also, and somewhere down the line I will be having magnesium here and silicon. Now this is clear. As you have to talk about the metallic nature, sir, as we move along the period, metallic nature decreases. As we move down the group, metallic nature increases. Now the idea was just to make the table. So who will be having the smallest value? Who will be having the smallest value? Sir, definitely along the period you find non-metals. And sir, what about beryllium? Ultimately, they are alkali and alkaline earth metals. And metals is supposed to have metallic nature. So beryllium, although it is amphoteric, but at the end of the day, it is a metal. And once you move along the period, non-metal exists. So who will be most non-metallic? Phosphorus. So obviously, phosphorus here also, here also, here also. After that, sir, silicon. So after that, silicon. So this option is gone. Yes, after that we'll have beryllium, because it is, although alkali metal, alkaline earth metal, but amphoteric, because size is also small. As size increases, metallic nature decreases. So then beryllium will come. And I guess because with comparing that beryllium only, it will be wrong, and you will be getting your answer as option 1. I hope it is quite doable. Quite doable. Just drawing of periodic table was necessary. Now, again a chemical bonding question, but I would rather say a very easy one. Acha, I guess all the options are not visible to you, so let me just yeah, I think now you can see this. Now you have to match list 1 with list 2. So sir, platinum complexes are always supposed to be square planar. So do you find any square planar complex? Yes sir. That means A is 3. So this is gone, this is gone. Now cobalt, sir, with plus with 6 coordination number, you may have sp3d2, you may have d2sp3 hybridization, but you may have unpaired electron, may not have unpaired electron, will depend upon the strength of ligand, but the geometry, but the shape will definitely be octahedral. So B is definitely 1, and by just checking that, B is definitely 1, so B is definitely not 4, na? So just by checking those two options, you are in your answer. That means option 4 is your answer. A very, very, very doable question. Ha beta. I mean, the questions which I am discussing right now, they are very easy. So I hope everybody has completed this. Chalo. Moving to the next one, guys. Yeah. Which one of the following is an ambident? Sir, again a very easy question. Sir, what they are asking? Ambident. The student has also marked it correctly, sir. The very famous thiocyanate. Thiocyanate is SCN. This sulfur is having minus, so this can also ligate. The nitrogen can also ligate. So whenever you have different side, but at one point of time only one can ligate, are commonly known as ambident nucleophiles. So option 4 will be definitely your right answer. Now, sir, you do not have to move your pen for such questions. People rush and read that NCERT formula. I used to tell in my classes, sir, what? Just go to your common sense. Oxygen making double bond. So this oxygen octet complete, this oxygen octet complete, yes. But this oxygen will say why, what I will do. So this will coordinate bond after formation is similar to a covalent bond. So replace it by a covalent bond. But if you do so, just remember the donor atom has lost 1 electron, the acceptor atom has gained 1 electron. So these are the formal charge. So formally what charge this oxygen has? 0. The question has marked this oxygen as oxygen number 2, this oxygen as oxygen number 1, and this oxygen as oxygen number 3. And now they're asking you that what formal charge is on second. So sir, it is 0. So 2 options gone. What the first one is having, sir, it is having +1 formal charge. So +1, option 2. I hope everybody have got the right answer, guys. Now, next question again from coordination chemistry. The type of isomerism you just have to predict. So platinum complex of the form Ma2B2. Sir, it is square planar, and square planar complexes can always show of the form A2B2. So this will be cis and it can have a trans. So this will always show cis and trans isomerism, which is nothing but geometrical isomerism. The student has also done it accurately. So that means A is definitely 3. So A is 3 as in 2 options are gone. A is definitely 3, so 2 options are gone. Now, B we do not have to check. Just let us check C because that will give us the tiebreaker. C is cobalt, 5 ammonia and NO2. Sir, NO2? Again, an ambident nucleophile because NO2, in NO2, nitrogen can also ligate and oxygen can also ligate. So this becomes an ambident nucleophile which shows linkage isomerism. So this can show linkage isomerism. So C is obviously 4. So third is your right answer. I hope everybody is correct. I hope everybody got it. Guys. Chalo. Moving to... Acha. Okay. That means, beta, I am done with the chemistry discussion. And yes, let me just give you a brief about the... Yeah. So let me just see which... Who will be coming for the discussion? Maybe biology. Or maybe Ambika ma'am or Shubhrat sir. Let me just confirm. And yes, by the time I'm just giving you my final word. Beta, chemistry was, yes, physical side of chemistry was requiring time for attempt. I mean, more than 50% of the questions I would rather say were moderate. But yes, if you talk of organic and inorganic chemistry, then those organic and inorganic were the area when you have scored to your maximum. Listen to me when I'm saying this. NTA has done this in the April and January attempt as well. They have done this. Physical, they have asked physical, I mean, just to be calculative, but in the JEA paper they have asked ionic and, I mean, very strong questions from electrochemistry, ionic equilibrium. But yes, I will rather say, but they are supposed to solve only 25, you are supposed to solve 45, so this is also very I mean, challenging. So yes, if you have invested and managed your time well, if you have invested and managed your time well, then I guess in for your chemistry, a mark of somewhere around uh, 80% efficiency. So let's say leaving 9 questions was maybe a cutoff. I mean, if you are planning for a government college, then you should have at least attempt 36 questions. So okay, so I guess we just have to just check. I'm just confirming, guys. Chalo beta. So at least let me give you an idea. Let me give you an idea of the questions that we had for chemistry. Let me give you a briefing, because yes, I have received that briefing. So at least let me share that analysis with you. I will be definitely the content that I've communicated right now, that beta, it is actually, acha, I am communicating in a different group or what? Yes. Hmm? Yes. I guess I'm communicating in a different group. Just give me a second, guys. Okay. Chalo beta. Let us have this detailed discussion for chemistry. It will definitely help you. It is definitely going to help you. Now, I would rather say, if you talk of chemistry, if I talk of chemistry literally, then a clear cut percentage which I can assign for chemistry will be, yeah, a clear cut percentage which I should say, that out of the 180 marks, that you were supposed to target for chemistry for your NEET 2026 exam, then you should have definitely not attempted 11.1% of the paper, where I would rather specially say those questions which involved from physical chemistry, which involved calculations. I would not say they were difficult, but yes, they involved a lot of calculations, and those calculations have taken could have taken more of your time, and that's why I would rather say that out of whatever questions I've discussed, 11% of the paper was hard. Talking about the when remember I used to say this: attempt the easy question in the first go and the medium in the second go. So after 1.5 hours, when you would have came back to your chemistry attempt, then you should have definitely attempted the medium level of questions. And if you have searched them properly, then around 37.8% of the questions were from medium. Yes, definitely they were belonging to very few from physical chemistry, mostly from inorganic, because IOC was very easy. And yes, obviously mostly from organic as well, but yes, or some organic questions were they were good, है ना? Chalo. Now, if I speak of the very easy questions, then obviously you can do the math, because obviously it will be more than 50% of the paper, beta. More than 50% of the paper was actually doable. You could have easily made it, you could have easily made your point. So my final word for the day, my final word for the day, sir, what is 180 into 51%? So somewhere around it is 90 marks. Now, what is 37? Let's say for case of simplicity, 10% of 180 is 10% of 180 is 18 marks. So 18 is 18 into 3.5. So just do 18 into 3.5. I guess 18 into 3.5 is somewhere around 63, I guess, na? 63. So if I add this, then this is 3 and 9 and 153 marks is the marks, beta, which should definitely land you to a, I mean, this much marks you should at least get. This is the minimum marks, minimum required marks for what? For a government medical college. Whatever score you are making, whatever score you are making, minimum this much of marks is a statistical thing that you should have got in your examination today. Then it will be a easy score, it will be an easy score to land you to a government medical college. Yes, obviously, if you try and reach more of 100% of this target, so obviously you may achieve so, है ना? So this is just a calculation which I am giving you, and with that, let us call it a day from my side, and let us call it a day from my side. So I guess Shubrat sir, okay, chalo. So do we have some more doubts, beta? Do we have some more doubts, beta? Guys, you can hit in the comment section if you have more doubts. Then obviously I can, or else I just, do we have any doubt now? The safe score which I have discussed, obviously, beta, it may have some variation, but yes, it will be landing around the marks that I'm giving you. So okay, so I'm ending because I guess Shubrat sir is ready. So I'm ending. Chalo. So Shubhra sir, over to you.
Hello, 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 hello. Hello.
All right, my dear friends. Hi, welcome back. Now you can hear me loud and clear, I believe. Hi, how was your physics exam? Quickly let me know. Be quick. How was your physics exam? And let's begin our discussion. Let's quickly begin our discussion of physics. Please note, obviously, we are solving paper code 12, right? Just like in chemistry and biology. Let's start. Yes, easy tha. All right, Sanya, Subhashri, paper was fine, paper was easy of physics. You guys were expecting that the 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 problematic part, the problematic subject is physics. This is what you guys were expecting, but in relative terms, definitely physics is easy. Most of the questions are either easy or medium difficulty level. Let's quickly start. This is the first problem, right? This is the first question, my dear friends. I'm going to read it for you because I can see question is slightly blurry. So I'm going to read it for you, and you guys have these questions. Here we have got 100 turn closely wound on a circular coil of radius 5 centimeters. So capital N is given as 100 and radius is 5 centimeter. All right, and then we have got magnetic field of 3.14. We have got magnetic field of 3.14 times 10 to the power minus 3 Tesla. This is the magnetic field, right? And this is due to a circular coil, my dear friends, right? So that means magnetic field formula can be written as mu naught I divided by 2 times into R. This is due to 1 turn, and let's multiply it by capital N. This is going to be the total magnetic field. Is it clear? Right? So they have told us magnetic field, and first they are talking about find out the magnitude of current and magnetic moment. So let's use the formula. Instead of magnetic field, let's write 3.14 into 10 to the power minus 3 is equals to, instead of mu naught, my dear friends, let's write 4 pi into 10 to the power minus 7 times total number of turns, which are 100, whole divided by 2 into radius is 5 and centimeter, so I'm going to write 100 over here, right? Pi can get cancelled out, and we have got current over here. So current will be, mind you, this is 10, so 10 times 10 to the power minus 3, this is 10 to the power plus 7, whole divided by, this is 10 to the power 4 and times 4. I believe it is definitely, you need to pay some attention, but if you are going to solve, you are going to get nearly 2.5 ampere, and we have got 2.5 ampere in two cases. Then you have to find out magnetic moment, that will be Ni into A. That means total number of turns is 100, current is 2.5, you can also write it as 5 by 2, and area is pi r square, and radius is, mind you, square of 5, and this is 10 to the power minus 4. This will nearly come out to be 2 ampere meter square. That is option number 1. If you are going to ask me what do you think about this paper, I am going to say paper is easy. The only difficult part lies in the numerical portion. Some of the questions are definitely numerically heavy, and that's what I have been telling you for so long, right? That please practice these types of problems in which you have to deal with powers, you have to deal with pi, you have to deal with squares of number, right? So first question was easy. Second question is absolutely easy. I have made one short also on this, YouTube short on elimination method, if you remember, right? And I have told you that whenever you are going to get questions on match the column, please try to do those problems in 10 seconds. Now let's suppose if you are planning to give NEET again, or if you are right now in 12th class watching how the paper is, then this is my advice to you. Please use these techniques in objective papers like NEET, in competitive exams like NEET. Now please see this question. Here you can easily see that when they are talking about this de Broglie wavelength, of course C is de Broglie wavelength, and we can use elimination method for C, because can you watch C? C is equals to 1 only in one option. C is it matches to de Broglie wavelength only in one option, that is option A, and we can directly match it, and of course then you can check all the other options, right? For example, as h mu is energy of photon, diffraction, this shows the wave nature of light, diffraction and interference, and Crompton effect is basically particle nature of light. So this is called as elimination method, and we have got multiple such questions in NEET paper. Moving on to the next problem, exactly the same question that we have done, I think in yesterday's class or day before yesterday's class, exactly the similar problem, my dear friends. This is reverse bias, this is reverse bias. The only difference is in my problem, the diode also had some resistance, right? In this, diodes are ideal. That means we are not going to take the resistance of diode into our account. Got it? Is it clear, my dear friends? Quickly let me know in the chat box. So this is reverse bias. I am sorry. This is reverse bias and this is reverse bias. That means we'll have 2 ohm and 4 ohm in parallel combination. Is that correct? So R equivalent will be 2 times 4 divided by 6, so this will become 3. That means 4 by 3 ohm is the R equivalent, and current will be voltage, that is 10 divided by R equivalent, that is 4 by 3. Clear? So this is going to be 2, this is going to be 5, that is 15 by 2 ampere is the correct answer. Now actually I was also worried for obviously for all of you, that if let's suppose NTA again do the same thing, that it shocks you with the first few problems, then you might not be able to do the rest of the paper with the same efficacy, right? But that's not what has happened right now. In this paper, the first few problems were very easy, very easy. So any student who has got the basic knowledge of class 12th, class 11th, NCERT, basic knowledge, this paper is very good to you, right? Very good for you. No problem at all. Now please see this problem. In this question they are saying the speed of light in vacuum is taken as unity if light takes 6 minutes 40 seconds time to reach the earth from the sun, the distance between the sun and earth is. You have to simply use the formula of speed is equals to distance upon time. Distance will be, speed is 1 and time is actually 6 minutes. That means this is going to be nearly 400 seconds and finally 400 meters. I'll explain it to you. 6 times 60 plus you have got 40 seconds, right? So this is 360 plus 40, that is 400. So 400 is going to be the correct answer. Is it clear? Right? Quickly write down in the chat box if you guys are following what I'm trying to say. In the next question they are saying the following plot show the variation of velocity with time of a ball thrown vertically upward and falling back. Which of the following plots is or are correct? All right, so that means over here we have got a ball and we are throwing it with a velocity of v. Obviously acceleration due to gravity is acting downward and it is constant. That means first velocity is, of course we are going to take the upward direction as positive, so velocity is going to be positive. It will decrease, it will become 0 at the highest point and then it will come back with a negative velocity. So option C is absolutely correct. All right, option C is absolutely correct. And if you remember from, I mean, if any student has done my formula revision sheet, I don't think there is any question out of those 3 lectures, right? We have done exactly this graph in that formula revision, if you remember, right? Moving on, next is in a vernier caliper, very basic level of vernier caliper question. They are saying that 20 vernier scale coincides with the 16 main scale divisions. That means 20 VSD is actually equals to 16 MSD, which means 1 VSD will be equals to 16 divided by 20. That is my dear friends 4 divided by 5 MSD. That means least count will be 1 MSD minus 1 VSD, and 1 VSD is 4 by 5 MSD. This will mean this is 1 by 5 and 1 MSD is actually 1 millimeter. So the answer will be actually 0.2 millimeter, and if they are talking about in centimeter, it will be 0.02 centimeter, right? So the correct answer is 0.02 centimeter. Is it clear, my dear friends? A very basic problem from vernier caliper. They have actually also not talked about, they have even not talked about errors, right? So this is very easy. Moving on, an AC circuit contains a resistance of 1 kilo ohm, fine, and a capacitor is of 0.1 microfarad and an inductor of 1 millihenry, and the resonance frequency is, so resonance frequency will be, my dear friends, omega is equals to 1 upon under root LC, or F will be equals to 1 upon 2 pi under root LC, right? That means, my dear friends, if I'll solve it over here, F will be 1 upon 2 into 3.14. This is a problem which is slightly numerical lengthy, right? The first problem, the seventh problem, is slightly numerical lengthy, and L comes out to be, L is actually 1 millihenry, that means 10 to the power minus 3, right? And capacitor is 0.1 micro, that means 10 to the power minus 7. My dear friends, I can, this is 10 to the power minus 10, I can write it as 10 to the power 5 over here, right? So this will become, if you will see, this will become 100, and I'll write one more 0 from here. So this is 2 times 314, and this is 10 to the power 4. So I don't believe this is extremely difficult to solve. I can rewrite this as 10,000 divided by 2 into 314, and this will be in kilohertz. This will be in kilohertz. I believe the correct answer will come out to be 15.9. 15.9, my dear friends. Can you please check? Be quick. Subhashri, everything's clear? How was your paper? Quickly let me know. Yeah, 15.9 exact, right? Clear, my dear friends? Moving on to the next one. Again, a basic problem from magnetism. We always discuss about this question. This question says, the given figure below shows a long straight solid wire of circular cross section of radius A carrying steady current I. The current I is uniformly distributed across its cross section. So that means, of course, it is not hollow, but it has current all over its mass, right, in the bulk. So the graph is going to be something like this. Magnetic field will be directly proportional till we reach the surface, and after that point magnetic field will be inversely dependent on R. That means it will move like this based on Ampere's circuital law. Ampere's circuital law is B integral of B dot dl is equals to mu naught times of I enclosed. Clear? Mu naught times of I enclosed. Very basic level of problem. Next question is, if you have remember, I was continuously saying that please solve Wheatstone bridge question. In the previous year also they have given this question, and in this year also, right? So the only the modification was that they have said that we have got a uniform metallic wire having a resistance of 4 ohm, and it is bent to form a square loop of ABCD. Fine. So that means this total resistance of 4 ohm is converted into a square, and that means 4A total side will be 4A. That means each side will have a resistance of 1 ohm. Is that clear? So that means this is 1 ohm, this is 1 ohm, this is 1 ohm, and this is 1 ohm. Now this is a balanced Wheatstone bridge. Clear? 1 upon 1 is equals to 1 upon 1. So we can remove this 2 ohm. So that means R equivalent of this will become 2, R equivalent of this thing will become 2, and finally we are going to have our final R equivalent will be 1 ohm. Clear? Finally the R equivalent will be 1 ohm. That means current will be voltage 2 divided by R equivalent, that is 2 ampere. So 2 ampere is correct answer for this. Alright. Clear, my dear friends? Alright, moving on to the next one. An unknown nucleus, again this is a numerically lengthy problem. Please see, an unknown nucleus has a nuclear density of this. That means nuclear density is given. Nuclear density is 2.29 into 10 to the power 17. And this is equals to mass of the nucleus, that is 9.9 into 10 to the power minus 27, and divided by volume, and volume is 4 by 3 4 by 3 pi r cube, and r cube will be, my dear friends, please remember it will be r naught cube times of a. Clear? Now everything is given, we need to find out the value of a. So a will be 19.9 times 10 to the power minus 27, and if you guys can see, they have given you the value of 4 pi, it is 12.56, right? Multiplied by r naught, they have given us the value of r naught, that is 1.2 into 10 to the power minus 15 and cube of this, and then, my dear friends, that's it. I think other than this, let's divide it by density, that is 2.29 into 10 to the power 17. Definitely, it is a lengthy problem, but mind you, solution, the conceptual, we have got no problem in conceptual part. Concept is absolutely clear. They have given us density, they have given us mass of the nucleus, and they are talking about capital A, right? If you are going to solve this, my dear friend, if you guys are going to solve this question, you are going to get option as 12. This is going to be nearly 12. All right, done. Moving on. Next question is saying that we have got a rectangular wire, okay, rectangular wire of loop 8 centimeter and 3 centimeter. So this is 8 centimeter, this is 3 centimeter, and we have got a small hole over here, right? We have got a small hole over here, and this is in magnetic field. This is in a magnetic field of 0.3 Tesla. Fine, it is in the magnetic field of 0.3 Tesla, and then they are saying the EMF developed across the cut, if the velocity of the loop is 2 centimeter per second in the direction normal to the shorter side. So this is the shorter side, right? And normal to the shorter side basically means something like this. This is the velocity, and velocity is, mind you, 2 centimeter per second. 2 centimeter per second. Clear? Velocity is 2 centimeter per second. So simply apply the formula E equals to BVL. E equals to BVL. Magnetic field is 0.3. This is 2 centimeter. So 2 centimeter basically means 2 divided by 100, and length is, my dear friends, 3 centimeter. So 3 divided by 100. Clear? So 3 3s are 9. This is 1.8. 9 2s are 18, and this is 10 to the power minus 4 volt. So 1.8 into 10 to the power minus 4 volt, that means option number 1. Clear? Right? Next is again a question from basic conversion of galvanometer. Please see, if you have any doubt, do let me know. Quick. Is it clear? My dear friends, come on. Everything's clear till here, right? Now in this question, they are saying that we have got a galvanometer. Its resistance is of 100 ohm, and the maximum current that it can withstand is 1 milliampere. And then we need to connect a shunt in such a way that we can actually convert it into a 10 ampere range of voltmeter, right? So out of 10 ampere, 1 milliampere will go over here, and rest of the current, that is 10 minus 1 ampere current, will go to shunt. Now again, please see, let's use the concept of parallel combination. Let's suppose shunt has got resistance S. So we are going to write 10 minus 10 to the power minus 3 multiplied by value of shunt is equals to value of galvanometer, which is 100 multiplied by 10 to the power minus 3. So value of shunt will become 100 times 10 to the power minus 3 whole divided by, my dear friends, in front of this 10, I can actually ignore this and reach to the closest answer, right? I'm using a concept of approximation, and then I'll get 0.01 ohm. So 0.01 ohm, that means option 1 is correct. I mean, in physics, I think option 1 were of many of the questions answers were option 1, right? Clear? So you can quickly get the answer like this. Moving on to the next one, 13th question. In this question, they are saying that in a YDAC, please write down in the chat box if you have any doubt. In the YDAC, using a monochromatic light of wavelength lambda, the intensity of the light and at a point on the screen where the path difference is actually lambda 1. It is lambda, okay, is K units. Fine. So first of all, you need to use two formulas. First is find out the path difference. Path difference will be 2 pi by lambda multiplied by phase difference is equals to 2 pi by lambda multiplied by path difference, that is 2 pi by lambda times lambda. So this gets cancels out and we have got delta phi equals to 2 pi, right? Next is the net intensity will be 4 I naught times of cos square phi by 2, and phi is, mind you, 2 pi and divided by 2, and this I net is also given to us. I net is given by k units. So this is k. My dear friends, cos pi is -1, that means this one is going to be equals to +1, that means k will be equals to 4 I naught. K will be equals to 4 I naught, and that means my dear friends, now let's read the rest of the question. The intensity of the light at a point where path difference is lambda by 3. So the new phase difference will become 2 pi by lambda times lambda by 3. So lambda and lambda cancels out. This is actually 120 degree. 2 pi by 3 is actually, sorry my dear friends, 2 pi by 3 is actually 2 pi by 3, and now we are going to actually divide it by 2, right? So this is going to be 2 by 3, 180 exactly, this is 60, this is 120 degrees. So now again let's write down the value of intensity. I net will be equals to 4 I naught cos square phi by 2, right? Cos square phi by 2. So this is going to become 120 degree divided by 2, that is 60 degree. So this is 1 by 2, so this will be 4 I naught times 1 by 4. So this is 4 I naught is actually equals to k, so the answer will be k by 4. Answer will be k by 4, that is option number 1. Moving on to the next one. So again we have discussed multiple times these types of formulas, both of these formulas in our previous few classes, right? Moving on my dear friends, in this question they are saying the magnitude and direction of the acceleration produced in a body of mass 5 kg when two mutually perpendicular forces 8 Newton. So you have got 8 Newton and 6 Newton are acting. So whenever you have got 8 Newton and 6 Newton, the resultant will be something like this, and obviously, and these are mutually perpendicular by the way, so it will be 10 Newton, resultant will be 10 Newton. Let's say this is angle alpha, so tan alpha will be perpendicular, that is 6 and divided by base. So this one is going to be, my dear friends, 3 by 4. So hopefully everything is clear. So 3 by 4 from 8 Newton, right? So 2 meter per second square. Let's also find out the acceleration. So acceleration will be net force divided by mass. So mass is 5 kg, so acceleration will be 2 meter per second square, and alpha, either you can say that alpha is alpha equals to tan inverse 3 by 4 from 8 Newton, or we can say from over here also, right? So this one will be 4 by 3. So 2 meter per second square and tan inverse 3 by 4. So option 3 is absolutely correct. Option 3 is absolutely correct. Let me know if you have any doubt. Clear, right? Any more doubts, my dear friends? Till here we have solved till 15th problem. This is the 15th one. Now in this question they are saying 5 capacitors of capacitances C1, C2, C3, C4 and C5. Alright, so C1, C2, C3, all of these capacitance are in series combination. These are in series combination, right? So that means if I am going to break it down, this one will become something like this, and all of these are of 10 microfarad, so this will become 10 by 4, and this is already 2.5. This is already 2.5, that is, this is already 10 by 4 microfarad. All right, and we have got a battery of 50 volt. Clear? That means both of them will have same charge because they are in parallel combination, and that charge will be equals to Q equals to C into V. Hopefully everything is clear, my dear friends. This is actually 2.5, that means this is going to be 125 microcoulomb. So this one will also get 125, this one will also get 125. This getting 125 basically means that all of these 4 capacitors will get 125 microfarad, right? So C equivalent is, by the way, C equivalent is going to be 10 by 4 plus 10 by 4, that means 20 by 4. This is going to be C equivalent, that is 5. So C equivalent is 5 microfarad, and Q is actually equals to 125 for all the capacitors. So that means option 1 is correct. As I am telling you, option 1 is correct for most of them, right? So if you wanted to use a Tukka method, then option way was the best option for you, right? Now let's move on. This is again a particular problem that we usually see in our notes. We usually discuss about this question. They are saying that in a meter bridge experiment, the positions of the cell E and galvanometer G are interchanged. We shall observe in the galvanometer. Before reading the options, everyone knows that a galvanometer works on the principle of a balanced Wheatstone bridge, right? So you have got R1, R2, R3, R4. Now my point is that this is it's going to be a balanced Wheatstone bridge. It's going to be a balanced Wheatstone bridge when R1 upon R2 is equals to R3 upon R4. I'm trying to say that let's suppose you interchange the position. That means let's write it as R1 upon R3 or R2 upon R4. Still equilibrium position will, still the balancing length will be at the same point. So balancing length will not change, but what's going to change is that the deflection can happen on either sides now, right? So let's suppose before interchanging them, as we were moving in this direction, deflection was happening in one direction. After interchanging, it can happen in the either directions, right? So correct option for this is that balance point does not changes. Balance point, no deflection, both right side and left side deflection can take place, right? So D option is absolutely correct. Next is the power of a crane which lifts a mass of 100,000 kg. So you have got 1000 kg over here. You have got 1000 kg over here, and we need to lift it up to a height of 20 meter. Up to a height of 20 meter. So work done will be mg 1000 into g into h, and if you are going to divide it by time, that is 10 seconds, you are going to get, my dear friends, power, right? So this is going to this will be actually 220 into 10 to the power 3 watt, that is 20 kilowatt. Now I know that they are saying that small g is actually equals to 9.8. That means answer will be slightly less than 20. Correct answer is 19.6. I have always told you, if options are far from each other, you can always use this technique. I'm so sorry, kilowatt, obviously kilowatt. Please see, I have also written kilowatt, so option 4 is correct, right? Kilowatt. Moving on to the next one, another match the column. This is what I was trying to say, that they have given multiple questions which are from match the column category, and always use elimination method for this. Kindly see that we can actually eliminate the option using option A. Option A is different in all the options, right? So A is Young's modulus. Now everyone knows Young's modulus is this, stress upon strain, F upon A times delta F upon A divided by delta L by L, right? So A is actually equals to 2. So A is actually equals to 2 only in fourth option, and hence fourth option is correct. This is how you can save time in match the column questions, right? In a concave lens, a ray of light emanating from the object parallel to the principal axis of the lens after refraction. So this is going to, it will appear to diverge from the first principal focus. Basics of diverging lens, that is concave lens. Nothing is needed in these types of problem. Clear? Everything's clear? Quickly write down in the chat box. This is a better problem. So can you please see from, I think from the last question that was good was, I think, this problem. So after 13th, we are having, we have got 7 easy questions, then we are seeing a moderate difficulty level problem, right? Now the point is, my dear friends, in this question, please understand, they are saying that we have got a thin wire of length L and linear mass density M. Linear mass density M, and it is bent into a circular ring. All right, it is bent into a circular ring. That means, first of all, please understand that the total length will become 2 pi r, and it is equals to L. That means radius can be written as L divided by 2 pi. Radius can be written as L divided by 2 pi. Also, my dear friends, everyone should know this, that moment of inertia about an axis passing through the diameter is actually written by this is actually mr square. I'm writing about mass, mr square divided by 2. That means moment of inertia about this axis will be mr square divided by 2 plus, I'm using parallel axis theorem, m, the separation between these two is of course radius, that is mr square. That means total moment of inertia will be 3 by 2 mr square, right? So finally the moment of inertia is coming out to be 3 by 2 m, but instead of m, my dear friends, we need to write down mass density, that is small m, multiplied by total length. So total length is L, multiplied by r square. Instead of r square, let's write L square divided by 4 and pi square. Hopefully everything makes sense, right? So after this, my dear friends, we will be left with 3m L cube divided by 8 pi square. So 3m L cube divided by 8 pi square, again option number 1. Again and again they are giving option number 1. Be quick. Clear? Everything sorted till this point, right? Moving on to the next one. Now they are saying that each side of a metallic cube of mass 5.580 kilogram is measured to be 9 centimeter. All right. Keeping the significant figures in view, that means please see over here you have got 4 significant figures, right? And then my dear friends, over here you have got 3 and over here we have got 1, right? So if you are dividing, we should give the answer in one significant figure. So that means either it's going to be option 2 or option 4. Clear? Either it's going to be option 2 or option 4. Now let's find out. They are talking about density. So density will be mass is 5.580 divided by, and then you have got a cube. So for cube, let's write 9 times cube of 9, and then please remember it is in centimeter. So 10 to the power minus 2 and cube of it. So this will become 5.580 times 10 to the power 6 whole divided by cube of 9. My dear friends, if you are going to solve it correctly, this comes out to be somewhat 7.654 into 10 to the power 6, right? So we are going to actually write, just a second, this actually comes out to be 7654 into 10 to the power 6, and we can again rewrite it as 7.654 into 10 to the power 3. And if we are going to make it to one significant figure, it will become 1 decimal place, then it will become 10 to the power 7.7 into 10 to the power 3. Please correct this. This is actually because we are dividing it. So this is actually 3 decimal places and this is 1 decimal place, 1 dp. So we'll give our answer in 1 dp. So that means 7.7 is going to be the correct answer. This is again a slightly, you need to pay some attention, else you could have also marked 4th option, right? Moving on. Again, this is a moderate level problem. In this question, they are saying that we have got an equation. Please see, we have got an equation y equals to 2 cos 2 pi. Let me multiply this 2 pi inside. So this will become 2 pi into 10 into t, then minus, you will have 2 pi into 0.0080x plus 2 pi into 0.35, right? So this is, my dear friends, if you are going to compare this equation with y equals to a cos omega t minus kx plus initial phase phi. So instead of omega, we have got, omega can be written as 2 pi f, and this 2 pi f is actually equals to 2 pi into 10. Actually, we don't require frequency, but fine. And then instead of k is actually equals to 2 pi by lambda, and this is actually equals to 2 pi times 0.008. That means 2 pi and 2 pi can get cancelled out and lambda will become 1 upon 0.008 or 1000 divided by 8, that is 125 centimeter. Now please also note down the unit. They are giving y in centimeter. Right? So this is going to be 125 centimeter or 1.25 centimeter. So 1.25 centimeter, but they are not asking this also. They are saying the phase difference between the oscillatory motion of 2 points separated by a distance of 0.5 meter. That means delta x is 0.5 meter, right? That is 1 by 2 meter, which means that phase difference will be 2 pi by lambda, and mind you, lambda is coming out to be, this is in meter, this is in meter. So lambda is coming out to be 1.25 multiplied by delta x, that is, so this one will become pi divided by 125, and this will become 100, right? If you are going to solve it, my dear friends, of course, if let's suppose if this was 1000, then we would have written it as 8, right? We would have written as 8, sorry. So we are going to write it as 0.8 radians, pi radian. So this is going to be the correct answer. Clear? If any student has any doubt, please note that you need to mind the units, and you just have to rewrite this in, you just have to compare it with the general equation. That's it. Is it clear? Any more doubts? Moving on to the next one. In this question, they are saying that a ray of monochromatic light is passing through an equilateral prism. Just a second, my dear friends. So exam day, so phone is constantly ringing. Now in this question, they are saying that a ray of monochromatic light is passing through an equilateral prism as shown in the figure. The reflected ray is parallel to its base. My dear friends, whenever they say that a refracted ray is parallel to the base, right, the first refracted ray is parallel to the base, always remember for R1 plus R2 is equals to angle of prism, we can write 2R is equals to angle of prism, or R equals to A by 2, and A is actually, angle of prism is 60 degrees, so this will become 30 degree. And that's also not what point is. Point is that if let's suppose this angle is 50 degree, and if this becomes parallel, then this angle is also going to be 50 degree, right? So angle of, if they are talking about angle of deviation, it will be I plus E minus A. That means 50 plus 50, 100 minus 60, that is 40 degree. This is going to be 40 degree. So these questions are very basic level 1 problems from all of these respective chapters, right? You just have to remember what happens when a refracted ray is parallel to the base. We can also prove all of these things, but you don't have to do it. You can simply move to this formula. Now this is a question which is slightly different, because in this you can get confused because of this waveform, right? Because of this waveform, but I've always told you, you have to choose the best possible options in these cases. So we have got a diode. Obviously in one half cycle it will behave as a reverse bias, and in another half cycle it will behave as a forward bias. So if this is a waveform, it will actually behave like a rectifier. So it will allow the current, it will allow the voltage to pass through it, it will allow the current to pass through it for one cycle, and for another cycle there will be no current. So best option is option 2. That's it. No more explanation is needed in this one. Now they are saying a resistor is connected to a battery of 12 volt EMF, all right, and we need to find out V. So E minus I into R. So E is actually 12 and I is 0.6 and R is actually 2. So this is going to be 12 minus 1.2. So this is going to be 10.8. This is going to be 10.8 ohm. All right, this is going to be 10.8 ohm. Next is the amount of work done to raise a mass of m from the surface of the earth to a height equal to the radius of the earth r. My students, please let me know in the comment sections if you do remember that we have solved exactly these types of problems from JEE mains in our marathon classes, right? Exactly same questions, my dear friends. So we need to lift a block from its surface to a height, and this distance is also r, right? This distance is also r from this surface. So from the center, obviously this is r, so this will become 2r. So work done is simply written as change in potential energy. So final potential energy minus initial potential energy. So final potential energy is, mind you, Gmm and minus Gmm divided by total distance is 2r minus minus plus Gmm divided by r. So into 2 into 2. So we'll get Gmm upon 2r. Let's do one thing. Let's write Gm upon r. Let's write small m and 2 over here. Let's write 1. Let's multiply and divide r. So we'll get something like this. This will become acceleration due to gravity on earth. So we'll get our answer as mgr by 2. mgr by 2. Fourth option. Clear? Right? Let me know in the chat box if you have any doubt. In this question, they are saying an electric heater supplies heat to a system at a rate of 100 watt. Okay, so if you are supplying heat, Q will be positive. We are going to use the formula dQ is equals to dW plus, sorry, du plus dW. So 100 is equals to system performs a work at a rate of, okay, work at a rate of, and we need to find out delta U. Okay, fine. So this is actually 75. So delta U will be actually equals to 25. What else I can say about the difficulty level of paper? Tell me, you tell me. What can I say? I'm really happy after seeing this paper, thinking that all of my students and in generally all the other students also, they are not going to feel that again that panic with physics. But please, for all the, I mean, all the students who are going to give NEET next year, do not take this for granted. Keep your preparation according to the previous year standard, right? NTA can actually behave sinusoidally, right? So this year again they have given an easy paper. Again next year they might give a difficult one, who knows. A room heater is heat rated, abhi this question is again from a very basic concept. 400 watt, 220 volt, that means rated power is square of V square of rated potential divided by resistance. That means resistance will be square of 220 divided by 400, right? And then if voltage drops to 200, then the power will become, so if it is drops to 200, so 200 times 200 divided by resistance, which is 220 times 220 times 400. Solve this particular value and you will get you will get your answer. My dear friends, for this you do the calculation. Calculation is also not very difficult, mind you. Correct answer is option 2, right? Correct answer is option 2. Clear? Any more doubts? In physics you are getting 170 plus 176 plus. That means only one question is wrong. Very good. Good. Next is, all right, this question is also a nice problem, a moderate problem. New question, you won't be able to see the last option. No issues, I'll explain it to you. When a ruler falls vertically, 5 different person catch it with different reactions time. Person A, person B, person C, person D, person E. My dear friends, the point over here is if anybody is going to drop from a particular height, this S can be written as half AT square. That means S will be directly proportional to T square, right? Now, based on the order of their time, you can arrange. Maximum time is for B. This is maximum, so B is for maximum, and then is 0.22, and then you have got 0.21, then is E, and then after this you all can do it. After it is A, and then you have got D and C. So I think correct option is B, E, A, D, C. For 38th, option 3 is, just a second, between D and C na. So D will come first. Absolutely. Yeah, D will come first. Let me just check this. Sorry, so this is actually C. I have written D and this is C. So fourth option is correct. Fourth option is correct. Alright, fourth option is correct. Clear. Consider two uncharged capacitors of equal capacitance 200 picofarad. One of them is charged by a 100 volt supply and disconnected. Now this capacitor is connected to the uncharged capacitor. The amount of electrostatics energy lost will be. So the amount of energy loss is simply written by 1 upon 2 C1 C2 divided by C1 plus C2 times V1 minus V2 square. Right. So obviously V2 is 0. We'll get 1 upon 2 C1. C1 is both are 200, and by the way these are picofarad, right? So this is going to be 10 to the power minus 12, 10 to the power minus 12, and over here it will become 400 into 10 to the power minus 12. Let's cancel out these two. Clear? Let's cancel out this. Let's cancel out this, and we'll be left with, please see, we'll be left with, we have got 10 to the power minus 12 and I have got actually 200. Anything else is remaining, my dear friends? I don't think so, right? Yeah, I'm sorry, V1 square also. So V1 square will be, V1 is 100, so the square of 100 will become 10 to the power 4, 10 to the power 4, right? So this will become 10 to the power 6, this will become 10 to the power minus 6. So we have got 2 by 4 into 10 to the power minus 6. That is 1 by 2 or 0.5 into 10 to the power minus 6. 0.5 into 10 to the, B option is correct. Clear? Moving on. Okay, Savita, 11th standard student, while conducting an experiment to determine the effective length of simple pendulum. Okay. All right, and then they are saying that notes down the data to complete 30 oscillations. To complete 30 oscillations, the time comes out to be 60 seconds. So for one oscillation, it will be 60 by 30. That means 2 seconds. It will be 2 seconds, right? So if time period is 2 seconds, that means it's a seconds pendulum. That means length will be of 1 meter. You don't have to solve questions like this. Length will be 1 meter if gravity value is equals to 9.8. That means if you are performing this experiment on the surface of Earth, then length will come out to be 1 meter. All right, moving on to the next one. Then they are saying that the peak value of an alternating current is 5 ampere. All right, so peak value is 5 ampere, and then they are saying the frequency is 60 hertz. How long will it take the current to go from 0 to the maximum value? So how long will it take? So my dear friends, please use the simple things like omega is equals to theta divided by t, or t equals to theta. Theta is pi by 2 and omega is 2 pi into f, and f is, mind you, 60. Right, so pi and pi gets cancelled out. This is 1 upon 240 seconds. 1 upon 240 seconds, that is option 1. 34th question is saying in interference and diffraction, the light energy is redistributed. If it reduces in one region producing a dark fringe, it increases in another region producing a bright fringe. This statement is correct, right? This statement is correct. As there is no gain or loss, this is not a statement. Now there is a statement. As there is no loss or gain of energy, these phenomena are consistent with the principle of conservation of energy. This statement is also correct. Diffraction and interference are characteristics exhibited only by light waves. No, it is also exhibited by sound waves and other waves, right? So this is wrong. Sound waves, interference, we have talked about interference of sound waves. We have talked about interference of wave on string. So this option is also wrong, my dear friends, right? This statement is wrong. Clear? So correct is A is true, B is false. A is true, B is false. Moving on. A box of mass 15 kg is kept on the floor on stationary trolley. All right. The coefficient of static friction between the box and the trolley is 0.12. All right. Keeping the box in stationary state over the trolley, the maximum acceleration with which the trolley can be moved horizontally is. All right. So please try to understand this question. They are saying that we have kept a box of 15 kg floor of a stationary trolley. So we have got a stationary trolley like this, and we have kept a block over here. Now with what maximum acceleration it can move so that block stays at rest. When this trolley is going to move, there will be a force will act on it, which will be equals to ma, mass of this block times acceleration of this trolley. Now this should be balanced by the limiting value of friction, right? So that means we are going to write m into a equals to, instead of mu, I'm going to write m into g and times mu. So m and m gets cancelled out. Acceleration will be 10 times 0.12. That means answer is 1.2 meter per second square. 1.2. Fourth option is correct. All right. Moving on to the next one. The sum of kinetic energy and potential energy of simple pendulum bob is 0.02 joule. Fine. That means total energy is 0.02 joule. Then they are saying the speed of the simple pendulum bob at equilibrium position is approximately. My dear friend, at equilibrium position, potential energy is 0. Potential energy is 0, and we have got kinetic energy is equals to half m v square, and that will be equals to 0.02. That means total energy will exist in the form of kinetic energy only at the equilibrium position. Clear? So that means v square will be 2 into 0.02 divided by mass, and mass is, mind you, 20 gram. So 20 gram means, let's multiply 1000 over here. So we are going to cancel out these two zero, this 0, we are going to cancel out this. V will be come out to root 2 meter per second, or 1.414 meter per second. 1.414, that is option number 1. Right? Moving on to the next one. 4 statements are given. The volume of the nucleus is proportional. So volume of the nucleus, just now we have written, it is actually 4 upon 3 pi r cube, right? So r is actually r naught, and this is a. So it is actually directly proportional to a, not a raised to the power. It is actually directly proportional to the volume of the nucleus. Fine. It is not directly proportional to a by 3. It is directly proportional to a. This is correct. The difference in mass of an atom and its nucleus is called mass defect. This statement is absolutely wrong. The difference in mass of nucleus and its constituent is called mass defect. This is correct. So statement B and C are true. Is that correct? Statement B and C are true, and A and D are false. Option 4. Next. The angular speed of a flywheel is increased from 600 rpm to 1200 rpm, right, in 10 seconds. They are talking about in seconds, so simply write, instead of 600 rpm, right, 600 divided by 1 minute. If you are going to convert it, we can write it as 60 rps, right? So the average angular velocity will be omega 1 plus omega 2 divided by 2. That is 60 plus 120 divided by this is in minute, my dear friends, right? This is in minute, and we want to convert it into seconds. Clear? So this will become 2, and just a second, hopefully everything is clear. This, we are going to rewrite this as 600 revolutions per minute. So we have got minute over here, right? So I am going to actually rewrite this thing as in seconds. So this will be 60 seconds. That means this will actually become 10 rps. Sorry, this will become 10 rps, and similarly 1200 rpm will become 20 rps. Now everything's fine, my dear friends. Now you can apply that. Average angular velocity will be 10 plus 20 divided by 2, that is 30. 30 divided by 2 is 15 rps. And if you have got 15 rps, and then they are saying the number of revolutions taken in 10 seconds, then again use the same thing. Theta will be omega times t. So omega in rps is coming out to be 15, and this is 10. So answer will be 150. That is option D, 150 revolutions. Clear? Now they are saying that a submarine is designed to withstand an absolute pressure of 100 atm. All right. So absolute pressure of 100 atm. So 100 atm can be actually written as 100 times 10 to the power 5 Pascal is equals to, we'll have 1 atm of course plus rho gh. kindly understand this concept. You have got a submarine, let's say somewhere over here. So the absolute pressure at this point will be P naught plus rho gh. So P naught is atmospheric pressure and this is rho gh, and this is equals to the maximum pressure that it can withstand, right? So that means H will be actually equals to, this will be 99 into 10 to the power 5 divided by density, which is 1000, and times gravity, and gravity is, I believe they have said us to take it as 10. So this is 3, 4. So this is going to be 990 meter. 990 meter is the correct answer. All right, my dear friends, clear? Any more doubts? Again, this is another match the column. This is what makes physics paper easy. Lots of match the column problems. Definitely gamma rays will be created by radioactive decay of nucleus, right? Visible light is actually when electrons in atoms emit when they move from a higher energy level to a lower energy level. This is absolutely true. If you are going to closely see again in option C, right? Option C is actually, option C is equals to 2 in third, in fourth and in first, fine. And then visible radiation is equals to, that is B is actually equals to 1 only in option 1. So this is going to be option B is equals to into option 1. So this is correct, and C is actually equals to option 2 in this one. Now we have to check for microwave, right? So my dear friends, the correct answer for this one is going to be A will be actually equals to 4, that is option number 4. Clear? So for 40th one, please kindly note, for the 40th one, my dear friends, option 4 is going to be absolutely correct. Clear? Moving on to the next one. Inside a conductor, the electrostatic field is 0. This is true, and we have to find out the correct statement. So A is correct. Electric field at the surface of a charged conductor does not depend on the surface, on its surface charge density. This is wrong. The interior of a charged conductor can have no excess charge. This is also true. At the surface of a conductor, the electrostatic field must be, this is also correct, right? The electrostatic potential is 0 everywhere. This is wrong. All right, my dear friends. So A, C and D only. A, C and D only. This is correct option. For a metal of a work function 6.6 electron volt, which of the following wavelengths of incident radiation does not give rise to the photoelectric effect? All right. So first of all, let's find out the energy. Energy is actually 6.6 electron volt, and let's use our shortcut formula 12400 divided by lambda. So lambda will be 12400 divided by 6.6. If you are going to cancel out 10, this answer will come out in nanometer. And my dear friends, if you are going to check the answer for this one will be 12400 divided by 6.6. It will be around 188 nanometer. That means any wavelength which is greater than this cannot perform, will not be able to perform photoelectric emission, right? So 150 is less, 50 is less, 100 is less. Answer is 200. This is the threshold wavelength. So if you want to select any wavelength, select less than this. Is everything clear, my dear friends? Be quick, right? Moving on to the next one. When the forward bias voltage across the PN junction diode increases above a certain threshold voltage, the current in the diode current increases significantly. This is absolutely correct. The current in the reverse, this current is called reverse saturation current. This is absolutely wrong, right? In forward bias, we never talk about reverse saturation current. So statement A is true, statement B is false, option 2. My dear friends, last question quickly. All of those students who have attended these sessions of me, Ambika ma'am and Gaurav sirs, do let us know in the comment sections how many of the questions were exactly similar from the marathons and from all the practice sessions that we all have taken, right? Paper was very, very simple, my dear friends. In these questions, again, they are saying that a flask contains argon and chlorine in the ratio of 2:1 by mass. Temperature, all right. So we all know that if they talk about VRMS, my dear friends, right, if they talk about VRMS, it is actually inversely dependent to the mass. That means V of argon divided by V of chlorine will be under root of mass of chlorine, that is 70 divided by 40, and answer will be root of 7/4. So that is root of 7 divided by 2. This is the correct option. All right, dosto. So this is all from my end. Now I have another live also. So hopefully your paper was good. If let's suppose still you felt some problem this year, then do not feel demotivated. Plan your year ahead, whether you want to take a drop or you want to get into college. We are going to discuss all of those things separately. So please be alert. We are going to keep on posting things on this channel. We are going to keep you informed how you should proceed with your with this year, right? Paper was good, paper was easy according to me, and it's not about me, paper is definitely easy. So we are going to plan the future things in coming days, right? So that will be all. Thank you so much. Bye bye, take care, bye.